spirits are as high as it was on the first day. But I'm here to break your spirits and tell you that whatever you have learned, you might have to unlearn quite a few points, because there are challenges. There are challenges in every aspect of proteomics, and just like any technique, it's not foolproof. We had discussed it in briefs while we were discussing sample prep, while we were discussing about max quant analysis, but now I will discuss in even greater detail. But what are the specific challenges in proteomics as a whole? Specifically, what are the challenges that we get during data analysis? So first up is sample prep, and I told you, although it is not related to the instrument, it's always, remember, garbage in, garbage out. So an instrument is only good as the experimenter, right? A Ferrari in the hands of Michael Schumacher is incrementally better than perhaps in my hands. So what are the challenge? So there is a risk of experimental failure, and this failure might come because of incomplete cell lysis and protein solubilization. So the premise of proteomics is that we are solubilizing proteins, and these proteins are then further digested into peptides or proteolyzed into peptides, and these peptides then fly in the mass spec and we get the results. But what if your very first step fails? I think a few of you at least who are sitting in the audience would have felt that there are failures. Jinal would have felt that. Shambhu has felt that, and I know it firsthand. So sometimes proteins won't just solubilize, or specific proteins that you want to study won't lyse. So all this becomes a challenge. So since we are not getting into the detail, because every problem, every experiment will have its own unique challenges. So before you start an experiment, do read up about the biochemistry of the component that you want to. If you want to work on cell surface, it will be different. If you want to work on something else, maybe a nuclear fraction or a mitochondrial fraction, so these things might have challenges. Another big challenge is contamination during protein handling, during sample handling. You see, we are ourselves very rich in protein. We have keratin flying all around, and keratin is the most favorite contaminant. And almost every mass spec data, if you analyze it, you would find keratin. There is no way of avoiding keratin until and unless you are perhaps in a very clean room wearing a complete PPE overall, the ones that they use in BSL-3 facilities. Else you would have keratin, but somehow people have found out methods to avoid keratin. But at the same time, you should also take precautions while you're preparing your samples. Suppose you are asked to fill up a tip box. Well, many people have this tendency to hold it by hand and put it in the tip box. The moment you hold a tip with your hand, you have introduced a new protein, keratin. And due to the homology, and since most of us work on animal cells, keratin will come up again and again. Detergent interference. We had talked about it when we were preparing that we need to avoid some detergents like NP40. We need to avoid it because what they do is they affect downstream analysis. They will intensively reduce. And how did they do that? They reduce the column performance, the LC column, the C18 column that you would be allowing your peptides to pass through. If detergent is present in that, you are more likelier to have a problem where the column performance will be compromised. Ionization is also getting compromised. So this entire premise that the peptides will fly, but peptides won't fly if they have detergents along with them. And even very small trace amounts of a detergent can suppress ionizations and signals. So you need to be very careful. So if you get into a greater detail, first reason that we discussed, that why is there incomplete cell lysis and protein solubilization? So if you go to this first point, so we'll now get deeper into it. So protein variability. Now when you're talking of a cell lysate or a tissue lysate, usually we would like to believe it's something that is very uniform, right? But we all know cells are not uniform. Every protein has its own hydrophobicity, its own character biochemically, right? And they are again complex, but sometimes with a lipid, sometimes with a carbohydrate, sometimes with DNA. All these will impact how it is getting solubilized. And for that, you have to really check for the specialized methods available. Whenever you plan a proteomics experiment, go back to the literature and see for this specific type of protein that you want to interrogate. Suppose you want to work on plasma membranes, a membrane protein. What is a way of enriching the membrane fraction? How was the best way to get enrichment in the membrane fraction? Suppose you want to work on nuclear protein or mitochondrial protein. I myself, I had a project on mitochondrial proteome, and the biggest challenge was getting clean mitochondria, because you need specialized ultracentrifugation methods where you would get a very clean pellet of mitochondria. So you have to set up density gradient centrifugation with Perkol gradient so that you get a clean band of mitochondria, and then you have to again separate it, but contamination will always come, and that will impact your signal intensity, right? We know because there might be some abundant proteins that might again interfere. So you wanted mitochondrial fraction, but you took a bit of cytosolic fraction as a contaminant. For that experiment, cytosolic fraction, although a protein, is a contaminant. So these are the challenges. Another is, now if you think that you will increase the number of steps, you are increasing your loss. Always optimize the number of steps, especially for proteins if your sample availability is poor. You are working with a histopathologist on cancer who will give you FFP tissues, and those tissue, they will give you maybe a few shreds of paraffin sections, and you want to extract proteins from them. Of course you can do that, and of course your high resolution mass spec can work with as little as 500 nanograms. But fact of the matter is, to reach 500 nanograms, you might have to do 5 or 6 or 10 steps, and these 10 steps might end up with only 150 nanograms. So this becomes a big challenge whenever you think that your sample is very complex and you have to add more steps. Remember that each step, none of the steps are 100% efficient. Efficiency is only around 50 to 60%. So you're losing out 50% of your sample with each step. Of course, it is time consuming and labor intensive. So how much time do you have? How many samples do you have to run? Suppose you are running a big clinical project where you have to go for 2,000 samples and you have 2 years to work with. That includes acquisition of samples, that includes preparation of samples, running the proteomics experiment, analyzing the data, coming up with a solid hypothesis. Can you afford to have a step that itself takes 7 days or 4 days? So there you have to make some compromises. Can we go for a step that can be scaled up? You might give up on a few proteins. So as I said, nothing is foolproof. The more you know a technique, the more you know how challenging it is, what are the pitfalls. And when you are designing an experiment, it's very important you know the do-nots rather than the do's. Reproducibility. All these difficulties will pose another challenge, that is, how reproducible is your method? How reproducible is your method? The more the number of steps, the more the human intervention, the more errors that you will put. Even if you work with maybe 5% error, now calculate if you have 20 steps, how much error you have introduced. And this error, when you multiply with the fact that maybe there are 1,000 samples, or even 100 samples, this error margin becomes so big that now your data is no longer reliable. You have compromised on data integrity, which is very important in science, that your results are reproducible. So this is a big challenge. So how to deal with it? Of course, we have discussed this, that you can use Rapidgest, Protease Max. So these are all companies, proprietary detergents that are compatible with the MS. Also, you have to check for phthalates. So when you are using a mass spec, so something that we always use is plasticware, right? Plasticwares leach compounds like phthalates, which will then go downstream and overwhelm the mass spec. So always make sure your plasticware is compatible with the mass spec. And how the one way of doing it is resourcing your plasticware from quality manufacturers. And if you know a lab who is doing mass spec, ask them what is the plasticware that you're using. So the problem is so vast that if you go for multi-center projects, they all source their plastic from one vendor with one single batch number. So even before the project starts, they know that I would require maybe 100,000 tips. And across 5 centers, I would need 500,000 tips over the next 3 years. They will purchase all those tips and ship it to the individual centers. Because even the best manufacturer in plastic, so we live in a globalized world, right? We do not know from which source who is coming, right? A simple thing, maybe this laptop, it would have components from all across the globe. The silicon is coming from somewhere, the plastic is coming from somewhere, the LED screen is coming from somewhere, the gold interfaces, that gold might have been mined in Australia, maybe in India, maybe in South Africa. You do not know. The battery components, they might be coming from different places. But over a batch, it is always same. So that's why you have to be very sure, especially when you're planning a big experiment, that all things come as a single batch. And especially if you are planning a multi-center experiment, again, all things should be coming from one batch and then distribute that plasticware and even chemicals to all those centers, so that you reduce the variability that is induced just by human intervention, or sometimes because lack of knowledge of the supply chain. Another, of course, is, as we discussed, abundant protein depletion and well as contaminants. So we have already discussed that abundant proteins like serum, albumin, they are so much in amount that when you do a mass spectrometry analysis to understand what are those proteins that are impacting your biology, the only protein that will seemingly impact your biology is albumin. Others will simply be masked by the noise, because the dynamic range of an instrument is limited. And this limited dynamic range, just hold on. मैं आपका नंबर एक स्टूडेंट से शेयर कर रहा हूँ. One thing is immunoaffinity depletion that does is it may unintentionally remove other proteins. Now suppose your biomarker had a very strong binding affinity with albumin. Now you have used a column that by immunoaffinity it is removing albumin. Your biomarker is attached to albumin. What are the chances that your biomarker will still be in the mass spec? We had discussed protein-protein interaction, right? So we always assume that when we are doing an immunoaffinity of the top 14 proteins, we are removing those proteins. We are only removing those proteins. Sorry to break the truth, we are not removing these proteins. We are removing those proteins and all those proteins which have a strong binding affinity with the top 14 proteins. Again, one more big challenge. And this is completely unintentional. This is not our intention, right? When we design the experiment, we always think that we are removing the top 14 proteins. These are our abundant proteins and we want to make it okay for the dynamic range. But ultimately what we end up doing is we are also removing those interactors. Next, protein contaminants, we had discussed this. Keratins from skin and dust. So most of the dust you see is the skin we continuously shed. We are shedding skins like we are snakes on a daily basis. So we shed skin every day and that skin ends up being the dust particle floating around. So if you are not working in a clean room, however hard you try, you would introduce some contamination. Enzymes from lysis and electrophoresis steps. So we are always doing lots of enzymatic proteolysis, but at the same time, when we add trypsin to a sample, there is no step where we deactivate trypsin. Of course, we do that, but do we remove trypsin? No, we can't. So I think we did tell you during sample prep that do not add extra proteins just for the sake of it. You can add, but there are limits to it, because then you are adding again something like you are adding albumin to your depleted plasma. So enzymes like proteases from digestion steps, this becomes a contamination that we can't help. There is always a contamination. What happens? First of all, the mass spec will analyze peptides from contaminants instead of target proteins. Especially true for DDA methods. When we are doing data-dependent acquisition, you see the more intense the signal, the mass spec will think, okay, this is an intense signal, let's delve deeper. And now your mass spec is wasting time on keratin, it's wasting time on trypsin. So it reduces the number of useful spectra recorded. Next, there are mitigation strategies. So you can implement an exclusion list. So when you design your mass spec flow, which we have not gone into detail because I thought first you need to understand the basic. So there are ways where you can limit the exclusion, make an exclusion list based on keratin. You can make an exclusion list of all the peptides that keratin can make. Based on trypsin, you can make an exclusion list that do not look into any M by Z that comes up with these values. That you can do. But limitations will always exist when contaminants are inherent to the sample. Now, suppose you're working on skin. Keratin is no longer the contaminant. Now, keratin is the protein you want to look into. So this will put a huge challenge. Again, you have to design a new method. So it's very important before you start an experiment that you understand the biology, you understand the limitations of the instrumentation. Another thing that we need is the conventional HPLC or the modern UHPLC fractionation. So always it is LC, MSMS. So the LC is that Either it's an HPLC or in some cases it's an ultra high pressure liquid LC. So what can we do? So what are the issues? Membrane protein solubility. So it requires specialization, special solubilizing agent like acid solvents, detergents, and cure drops. And these agents need to be diluted or removed from MS analysis. So membrane proteins pose a big challenge when we are talking of HPLC-based fractionation. So we want special acidic solvents. You see, we are adding 0.1% TFA, right? We sometimes add cure drops as well in the buffer. So these buffers try to maintain the solubility of the membrane proteins. Next, what you need to understand, the ionic strength and the buffer capacity. What is the buffering capacity of your HPLC buffer? What is the ionic strength of your buffer? Right? These things you have to always keep in mind. Next, pH response affecting the retention time and peak shape. Last time when we were yesterday or day before yesterday, when we were showing you peaks, even Professor Sanjeeva showed you chromatograms. So pH response, the pH of your buffer, if it is altered, if your buffer somehow pH is not properly set, again we will get a huge lot of noise. So that is one another limitation. Issues with this SCX and HPLC separation. So you are using a column and the peptide dilution is not okay in that column. Maybe the C18 material was not packed properly, or in the peptides, the way the peptides interact with the C18 material is getting influenced by the buffer and the elution is not proper. Especially important when you are going for unlabeled variation. So when you are doing labeling, you have challenges. When you are going for unlabeled proteomics, there this elution time and in fact this imprecise peptide elution can be a challenge. Next, peptides appearing in multiple HPLC runs. Now what if you are not running enough blanks? And what happens is your blanks and sample, they start to look same. Suppose you are processing 100 samples and you are in a rush or your column is compromised. What happens? Few peptides, they so strongly bind with the column that they are eluted repeatedly. So there, suppose 100 peptides bind. Peptide A, 100 peptides bind to the C18 column. When you are running a regular gradient, 25 will elute out, 75 will stay back. Now you run a blank, again 25 will elute, 50 are still staying blank, staying there. Now you run another sample, you add another 100. So now your column contains 150 peptides. This time maybe because there are more and 25% need to elute out, this time how much it will elute? Something around 37 molecules, 37 peptides. Now this builds up over time, and by the time you are in your separate, you are in your 20th or 25th sample, the peptide is consistently eluting from the sample, even in blanks. So important thing to understand here is you can avoid it by running proper blanks and also observing if the blanks are actually looking like the blank. So blank should ideally be a very straight line on a chromatogram. Complex samples. So when these samples are eluting out, the singly charged peptides, they are the fractions which will elute first. They will elute first and they will dominate the entire mass spectrum, the chromatogram. The di and triprotonated peptides are not easily trapped in the column. So this again impacts your data. Then there are technical challenges in liquid chromatography. Difficulty adjusting equipment for multiple parallel columns. Usually, in a lab, you would have a high column, maybe a C18 column. So multiple columns are present, especially those who have already worked in a mass spec environment would appreciate this fact. We don't work on single columns. But every time you change a column, due to lack of time, due to lack of precision, you don't change your instrument setting. So the same pressure is applied on a new column and on an old column. But the binding affinity of the C18 material after maybe 1000 runs have significantly changed. You should change so the new column would be significantly fresh, it can handle a high pressure, whereas the old column, which is kind of choked up now, you have to run it on a lower pressure, but we don't do it, because if you lower the pressure, immediately the chromatogram elution time will vary. So you have to be very precise when setting these things up. This is again a big challenge. It needs lots of labor, lots of time. Column cost and stability. So columns are usually very costly and they are not very stable. So you always have to have good money in your facility so that you can keep on buying new columns, and also new columns might fail. And that we have observed with Professor Sanjeeva's lab when he was working with 3 mass specs. We almost had around 25 columns failing in 8 or 9 months. 25 new columns. That entire batch had a manufacturing defect. And it's very hard to convince the vendor that you have done something wrong. Those who are working on antibodies, on Western blots, you would know. Suppose an antibody is not working. The first thing they will ask you, show us the blot. Then show us the protocol. Then they will give you an alternative protocol. Then you have to again work on it. Again you have to show us them the blot. So maybe 6 months will go in convincing the supplier that they were wrong in the first place. Then they will take, okay, we are setting up a replacement, and this will go through our finance section. So from the field scientist, it goes to the sales and finance. They are very hard to please people. They will again ask for a report. They will schedule an engineer visit. So for 1 month, or for 1 year, you keep on convincing, and after 1 year, they will try to reach a truce. Okay, so we supplied 25 columns, they were wrong, we understand the issue, but we cannot issue 25 fresh columns. We will give you 10 instead. Now you will start bargaining. From 10, you will say, no, not 10, 20. You will say, okay, not 20, 12. You say, no, okay, not 12, 18. And finally you bargain at 8, something like 15 or 16. Then they will say, okay, I am now setting it to our consumables department. They will deal with it. Now the consumable department will ask you for all the proof. That what went wrong, get a report from the field scientist, get a report from the finance, and effectively you have been pushed back by a couple of years. Else, if you have lots of money, you keep on doing the finding, apply for a fresh set of columns with a new vendor. You are going for a new vendor, you are not satisfied with the current vendor, they set in a new column, now you have to set in a new method. Right? So you have lots of cars, you are driving one model, you are shifting to the other model. At least initially for the first 1 week, you will be very skeptical how this car will drive. How does it accelerate? How does it brake? Try, if you have a bike, try riding your friend's bike. Its brake response, its throttle response will be significantly different from yours. Same happens with instrumentation. So what we need to do is need for advanced column technology and bringing every system at par, right? So that columns become instrument agnostic. When a column becomes instrument agnostic means whatever instrument you use, all the companies are making a similar type of column with similar type of settings. Very hard in a market where everybody wants patents. Even our universities want patents. Your CV should have a few patents. So whenever you patent something, you're hiding something. And the moment you're hiding something, it's no longer open source, and something which is not open source cannot be effectively copied. Next, the trade-offs between resolution and speed. So anything that is fast, you will agree. So if you have to cook a good food, it will take some time. But you have to have something very fast food, you can do it very fast, right? You are compromising on the taste because now you have to eat something. So you can have 2-minute noodles, but you have to cook a good wholesome meal, it will take you around a couple of hours. So if you have to compromise on speed, first then you have to compromise on your health. You know noodles is not good for you, but at the same time it's saving you time. So you decide, okay, let's save time today. That's what pharmaceutical and clinical labs do. They need to sacrifice resolution because the moment you increase the resolution of a mass spec, you have to run a longer gradient, right? So a gradient that we use in research labs can be as long as 120 minutes. Pharmaceutical lab does not have time, or they do not desire to change the speed. So what they need to do is, they will essentially sacrifice resolution, run shorter gradients, and somehow get the data. So whereas fundamental research, like suppose you're working on a pathway, and you need to have a much greater resolution, so you have to have a longer dynamic range, you need slow elution, so you will run a 2-hour gradient, and that gradient will give you much greater proteome coverage, because now you want to analyze a very complex mixture, you want to do some drug development, you want to identify a novel biomarker. So this is so I mean, you can say this is one of the issues with any experimental design. Even if you want to have to scan for a biomarker, once you have established a biomarker, you want to scan. You will just scan for that specific M by Z ratio, even if it is a orbit trap or a triple quad, right? You will perhaps run a PRM. PRMs are much faster. You can run it in 30 minutes. So thing is, we need to continuously improve upon these HPLC and UHPLC system. We can have better resolution in mass spec, and we are doing it. Like as I said, 2 years back, people were getting around 800 or 900 proteins in plasma using DDA. Then you move to DIA, you get 2000 proteins. You move to a higher spec timsTOF or maybe an orbit trap Astral, Thermo Fisher Astral. Then you can push the number of proteins identified, something around 4,000, 5,000, again depends on the method. And this is really required if you want to elucidate the biological mechanisms and you want to know the real story behind the disease development. So we have already discussed few things, but again I think it's good to understand what are the challenges in MS instrumentation. So mass spec is a very high cost, high maintenance instrument. The reason you won't find the mass spec in an average lab. You might find a real-time PCR, you might find a spectrophotometer, all these things. But anyone from a lab which has a mass spec of its own, anyone in this room? Perhaps no. Anyone who has it in the institutional central facility? 1, 2, 3, 4, 5, 6, 7. So that's the thing. Most of these instruments are capital-intensive instruments, costing crores of rupees, and they are very delicate as well. As I say, a small change in column might put things off. A small change in pressure, things will go off. So most labs require mass spec labs, a good high-performing mass spec lab, are run under 24-hour supervision. All the time, somebody is keeping an eye. You don't need to be in that mass spec room. You can use softwares like AnyDesk to be at home or while you are traveling. You can share the screen and you can have remote access. But at the same time, you have to look at it. If the pressure is falling, if the pressure is suddenly very high, if the mass spec has stopped giving you readouts, suddenly you are seeing even your samples are giving a straight line. All these things, and of course, troubleshooting and maintenance, extremely difficult. So we had three mass specs in Allahabad at IIT Bombay when I was doing my postdoc. The vendor was 2.5 kilometers away. The guys who sold it, their India head office was 2.5 kilometers away. And guess how much time they used to take to come to the office if there was a maintenance? How long would you take to travel 2.5 kilometers? 4 hours. They usually took 4 weeks. So you have to raise your complaint. Somebody will tell, okay, we are looking at it. Give us access. Are you sure this is the problem? Just like with any customer service. Once they have sold you their instrument, their customer service takes a back foot, then their maintenance division wants to sell you more spare parts. इसमें ये लगेगा, इसमें वो लगेगा. So that's the biggest challenge. Data complexity. So one more thing that is having instrumentation issues is excessive protein information can lead to false negatives and positives. As I said, we only assume that this is the protein based on its proteome, MIZ signature. So we match the signature from theoretical spectra with the practical spectra. But the thing is, this can lead to false negatives and positives. We'll come to discuss why. Unassigned spectra complicate the data analysis. So as I said, if you have too much of contaminants and all these kinds of unassigned spectra, the instrument will anyway try to make sense of it, because until unless you tell it not to look into something, instrument mass spec is actually somebody who will look into everything, right? A simple thing is you can take up the whole proteome data of the blood, right? Now, in today's world, we know that blood also contains some microbes, right? So blood contains healthy microbes, right? Maybe our gut contains microbes and they leak some proteins. Now, do you think when the mass spec is looking into the mass spectrometry, when they are looking into the data, do you think they're only looking at human-specific information? No, they are peptides. So any peptide, even those peptides that came from your hand while you are eating chicken burger, and then you forgot to wash off your hands, and somehow that chicken burger fell in your sample prep, a part of it, they will also analyze chicken proteome. So this takes and complicates. Again, instrument limitation. No single instrument can perform all experimental types. So if you want to do a really good quantitative proteomics, targeted proteomics, a triple quad is a better instrument. A triple quadruple. If you want to do discovery, a hybrid instrument like Q Executive from Thermo Fisher, it's a good experiment. But there is no thing such as I have one experiment, I have one mass spec, and now I can do all kinds of proteomics. You can only do bottoms up proteomics. For a top-down proteomics approach, you would need a different kind of mass spec, right? There are multiple types of mass spec. Since it was only focused on proteomics, we didn't go into those details, but you can look up. There are so many different types of mass spec, and each mass spec has its own strengths and flaws. Dynamic range, we have already told, they have a very short dynamic range, and that dynamic range might again pose a challenge. When we are talking of biomarker discovery, the biggest problem is the dynamic range and high complexity in biological samples. Biological samples by nature are very complex samples. You have lots of proteins. Take up a cell, think of the proteins in the plasma membrane. Are they hydrophobic or hydrophilic? They are both. The transmembrane protein, the cytosolic domain and the extracellular domain, they will be hydrophilic, but the transmembrane domain will be hydrophobic. So one protein, you don't know whether it is hydrophilic or hydrophobic. It can be hydrophilic and hydrophobic based on the domain. Now, these kinds of information can make the sample complex over and over again. And these complexities add up, and it becomes a big challenge for the mass spec to go deeper into it. Low abundance of disease-specific biomarkers. Usually, disease-specific biomarkers are usually very low in abundance. They are not very high because they are not constitutively expressed. So when you are taking a blood sample from somebody, this blood is like the sewage system of our body, right? It goes through every cell, picks up small proteins that are leaking into, and then it keeps on transporting those signaling molecules. So if I have a small cut in my hand here, even if it's a very deep cut, all those inflammatory molecules and proteins, what are the chances that if I have a blood draw here, it will be coming? The chances are very low. So I have around 6 liters of blood, maybe only 5 or 6 ml is actually having that information. Now, the moment it spreads down, it comes down thousands of times less in concentration. Now it becomes a challenge for the instrument because, again, we discussed that dynamic range is an issue. It will not pick things up. Instrument inefficiency, of course, we discussed that no instrument can fix. There is no one size fits all. Qualitative and quantitative proteome characterizations, again, remains a challenge. Now let's move on to what are the challenges on mass spectrometry data. The ones that, so after all these challenges, you get a data, right? You get a raw file that you can set up on MaxQuant for an analysis, or NMS Tracker, or Proteome Discoverer, depending on, or Spectronaut, depending on your instrument type, and depending upon your lab funding. So if you're a rich lab, you will use Proteome Discoverer or Spectronaut. If you're from a poor lab, you would perhaps use MaxQuant and NMS Tracker. Not true, but that's how things work. Some people have the money to subscribe, and those subscriptions are really costly. I think around 8 lakhs per year for Proteome Discoverer. One user. You can have only one computer in the lab running Proteome Discoverer. And suppose there are, and sometimes even rich labs can't afford it for the very simple reason that they are having 5 or 10 mass specs, and it's a big team of 50 people working. So you cannot take 50 individual subscription. And the group subscription is also inordinately high. And if something free and open source can solve the problem, why should I spend money? You see, scientists are very challenged when it comes to money. And nowadays, there was a time when you used to get money and you used to publish free. Now you have to even to publish, you have to pay money. So this entire industry is about paying others money. So once the data is generated, the biggest challenge is peptide identification. Why? We have already discussed. We identify peptides based on the M by Z data of known proteins and theoretical fragments. Two things you have to remember: known proteins and theoretical fragments. That means a mass spec and a software like MaxQuant is limited by two things. Number one, it can only look at proteins which are already known. The reason I am telling you this is we have a lot of unknown proteins, right? Professor Sanjeeva in the very first lecture told us that a significant portion of the proteome is unknown. So we are working on an assumption that whatever we are doing, matching them by Z data of known proteins and theoretical fragments. So this is an assumption that my sample based on my database can contain only about 8,000 proteins. We are assuming this. It might contain 80,000. Is there any way to know? No. Because use all protein coding genes are known and well admitted. It's a big assumption. Sometimes you can read like databases like NCBI, RefSec, and UniProt KB. We usually take data from RefSec and UniProt. A big challenge again. What is the reference sequence? It's just one representative sequence. Right? If I start sequencing the genome of every student here, all of us here, do you think we'll get 100% match even for a gene like glycerol gap DH or beta-actin, something that is so important? You will have single nucleotide polymorphisms that will change your proteome. Your beta-actin and my beta-actin might be very similar, but not exactly similar. And since it is not exactly similar, it might have something analogous to RFLP in DNA. It might have that the trypsin cut sites have been altered. The peptides that your fragment will generate, your proteome will generate, and then the peptides my fragment will generate are different. But the mass spec does not know that. Max quant does not know that. That becomes a challenge. Again, a big problem is the PTM, or the post-translational modifications. Now, PTMs are invisible at the level of the genome. But what PTM does, it changes the mass. It changes the mass by changing of the mass, the M by Z data, the M by Z will also change. You agree? All of you agree with this part, right? If I change my mass, the mass by charge ratio changes. So PTMs and genetic data cannot tell me whether the PTM is probably there. So again, these modified peptides are thus absent from the database. So this again complicates the proteomic data analysis. So this is again one pitfall. So see, this session is all about pitfalls to make you sad. How do you do this? So incomplete. So another problem is this de novo sequencing and proteogenomics. I think we had discussed in brief what is proteogenomics in one of the lectures that I was giving. So incomplete peptide fragmentation and low mass accuracy. So as I said, HCDs, or the fragmentation that happens at MS2 levels, is again not always complete. It's sometimes a chance factor. The better your instrument, the better the fragmentation, but sometimes again the instruments are not working at 100% efficiency. The instrument efficiency alters. So when there is alteration in protein, these efficiencies, there is incomplete peptide fragmentation and low mass accuracy. There have been recent software improvements, like there is something known as the GIAN, or the DIA neural network. So this is an AI-based tool that tries to correct for these changes. But again, there are limitations. What is AI? How do we develop an AI? These are models, and these models are developed on existing data. So last time Professor Graham had addressed a webinar here, and he said that I would rather use augmented intelligence than use the term artificial intelligence. What AI does is make things easier for you, but it can never be creative. It cannot make anything new. It only regurgitates what information has been fed to it, and it will perhaps do a better paraphrasing of the information. right? But something like extremely novel, extremely new, highly unlikely, at least at this stage. So AI will also be limited by the same challenges that we discussed, that the RefSeq database is primarily a RefSeq. The entire idea, when we talk that we all humans are different, we have a different genome, and then I tell you, we have a reference sequence. Beta-actin sequence is this. GAPDH has this sequence. We have been hypocritical. How can we know a GAPDH sequence for sure if we are all different? It is very similar, but not the same. Now there is a solution to it, and the solution to it is you use proteogenomics approach. You sequence everybody's genome, and from that genome you do a 6-frame translation, and now you have all forms of protein in your database. But now you have made your database 6 times larger and your analysis sufficiently cumbersome and more processor intensive. So to solve one problem, you have created another problem. Another problem is amino acid alignment. Many peptides are not unique. Last time, I think in the Max Bond lecture, I would have shown that there is something known as a unique peptide. Right? A peptide that is unique to a specific protein, and then there are lots of peptides. They are not unique. Sometimes there is high similarity of amino acid sequence between species. And if there is a high similarity, you might put your input data as rat, and then you will have 4 human proteins suddenly coming up in your data. Shambhu would agree. You started off with rat, and suddenly your analysis is showing that you have 4 human proteins. The reason is they are so similar. Also, closely related species will have about 99% amino acid alignment, and distance related species have 80% alignment. So what it does is, if you want to analyze your data, suppose you have a data set where you want to separate, right? You are trying to work with a host-parasite relationship, and you want to check what are the host proteins, what are the parasite proteins. Now you know your proteomics is actually both having host and parasite proteins. But the fact of the matter is, sometimes you will identify a parasite protein just because it is very similar to the host protein. You might use 2 different runs on Max Bond, so in which you will first use a human database and maybe then a parasite database. But since they are having similar, what will happen is you will get conflicting results. You will see that there are lots of proteins which are actually not present in the host, but are actually present in the parasite. It will come up as a host protein and vice versa. Any impact on data analysis also difficulties in identifying homologous proteins. Now there are proteins which are extremely homologous, especially that belong to families. GPCR family is one good example. So there we have lots of GPCR family proteins. Now when we want to do a mass spec on that, just because the peptides that they generate and the unique peptides are actually less, you will end up getting lots of data that is complicated by the very fact that there has been a misidentification because the proteins are so similar to each other. Another is in quantitative proteomics. So if you want to do a reliable quantitative analysis, you would have to use systems like SciLac, TMT, iTrack, and these instruments, these reagents are very costly. You can do label-free approaches. They are very cost effective. One sample run would cost you around 10 to 15,000. But if the MS conditions vary, if the LC conditions vary, again analysis becomes a challenge. In the label-free method, the biggest challenge is the technical replicate. The same sample you run thrice, you will get 3 different proteomes. There will be around 80 to 90% match. But even then, each cycle you will identify 10% new proteins. So how do you go ahead? So you have identified 100 proteins every run, but out of that, only 90% are common to all the 3 runs. Each run has identified 10 new proteins. So suppose you want to identify a biomarker. How do you go ahead? The only way ahead is to remove all those 10 unique proteins in each of the run and only go ahead with those 90 that has been repeatedly found. Something I think Shambhu would have demonstrated in the metabolite analyst. Right? We remove, by statistical filtering, we remove all those proteins that are not in every run. Time, it's a big challenge. When you're doing a small project, it's fine. If you're doing a big project, a multi-institutional project, and there you are trying to do samples in the tune of thousands. And as of now, in India, nobody does that, but we have European labs and American labs who do that. Label-free means each run will take 2 hours, then a 1 hour blank. Effectively, you can run only 8 samples per day. The reason those instruments run every time. Due to the different physicochemical properties, the peptide behavior will not be uniform each time. Electrospray ionization is good, but how they are ionized by this huge potential difference, that ionization is random. Every time the same peptide might not get ionized. Again a challenge. So it also, for all these reasons, it hinders the accurate quantification. Now, since you think that these are the issues of label-free, let's go for labeled. And all my issues will be solved. Sorry. We don't have solutions in this lecture. We only have lots of problems. Okay, the lecture might get demotivating, but I have 8 more slides to go. So if you go for any of these labeling methods, and I think I told you at the very beginning, no labeling chemistry is 100% efficient. Efficiency is low, and sometimes these labeling methods, like something like SciLac, is only applicable to cell cultures. So if you want to work on a live mammal, you have SciLam, but SciLam has its own challenges. There you need the rat or mice to eat isotope level labeled food. If the rat is like me, it will eat more. But there are people who eat less. I will take up more labels despite being in the control group. And somebody who is a friend of mine who would eat less, they will take a much less label. Now when we are quantifying, there will be changes. Right? So, one more problem. So the next problem is false positives, false negatives, and unassigned spectra. So what is a false positive? Forget mass spec. What is a false positive? Right. So actually there is nothing, but you are shown that okay, you have this problem, or you have anything, like somebody tells you you are smart, but deep down you know you are not. So that's a false positive identification. But quite the opposite happens most of the time. Once you go for job interviews, you will get lots of false negatives where they will tell you, you are absolutely dumb, you are not suited for the job. But you know you are suited for the job. That's a false negative. And unassigned spectra, as we had I think discussed briefly, that there are, just because it is not available in the database, but it is there in the actual data, that spectrum is unassigned, right? Suppose I have a protein that is extremely novel, that makes me smart, handsome, intelligent, whatever. But that protein is only present. Suppose I am Superman or Batman, I can fly. Now I have that protein, but the proteome database at UniProt does not know that I have that protein that makes me fly at exactly sharp at midnight, right? So however costly may be your instrument, you won't be able to find the protein, because we are completely contingent on the database that we are using. So that spectra becomes unassigned, right? So persistent false negative identification, peptides and proteins may not be detected, and there are multiple reasons. Maybe the peptides cannot fly. Maybe the peptides by trypsin digestion ended up being less than 7 or more than 35. Can't detect. Physiological or pathological factors, lots of PTMs, modified peptides like phosphorylated peptides, acetylation, they will not match the database entry. You can't detect those as well. Rare PTMs like are often overlooked in searches. So if your PTM event is rare, it will again be overlooked. Unknown PTMs, there are a number of post translation modifications that we do not know yet. And since we do not know, just like AI, it cannot tell us that what we do not know, right? So this is a big limitation. So what you could do is you can expand the searches to include more PTMs. Like if you go to MaxFont and set it up, you can include all known PTMs. But the moment you do that, time increases. Then you have to have a really fast computer, something like 128 GB RAM, and you have to go to a server. You cannot do it on a PC, right? Powerful software, extended analysis, of course, more electricity. Since you're running a server, now you need air conditioning, and you need 24/7 power. You cannot work in a system where there is a power cut, right? Because there is a power cut, the entire process stops, you again start from scratch. So all these big labs where they do lots of big data analysis, their servers are latched on to UPS systems that can serve till almost like 12 hours. So suppose there is a grid failure. The first day I entered my postdoc lab at IIT Bombay, there was a grid failure. Entire city of Bombay, which usually has extremely good power, the grid failed, so there was no power, almost for like 20 hours. And everybody thought I would be a very bad influence in the lab. Artificial modifications during sample prep. So what can lead to false negatives? Methionine and tryptophan oxidation, cysteine oxidation, idiacetamide modification, acrylbide modification of cysteine. So all these kinds of small changes can change your amino acid mass, effectively changing the ratio. And since we are always matching with the theoretical spectra, my theoretical spectra was supposed to be here, right? My spectra is here. Now this becomes unassigned. It's only when there will be a match, and perhaps this should be the perfect match, even if this match is there or there, the computer can make those corrections, right? The software can allow something known that we know as a mass tolerance. We when we do some analysis, we allow a mass tolerance that this much shift you can allow. But a mass tolerance of this, a wider mass tolerance, is not acceptable, because then there will be, all the results will be false positives. So the moment your spectra becomes unassigned, that becomes a problem, and these can be a number of reasons why you can have these false negatives. Poor ionization efficiency, hydrophobic proteins and peptides. If your protein is hydrophobic, it is more often than not have a proclivity to not get ionized. It won't get ionized, and since it is not getting ionized, there will be a problem. Natural PTMs, glycosylations and disulfide bridges. So it will lead to mixed cleavage, something we know as the mixed cleavage. Mixed cleavage means there was a cleavage site. Since there was sulfhydryl bond, cysteine bonding, double bonding, this cleavage site was missed by the enzyme. Now you allow for one missed cleavage, but sometimes if there are bad sample prep, you might have 2 to 3 missed cleavages. It can also hinder fragmentation and then lead to poor MS2 spectra. So MS spectra will be hindered, because even the peptide, if it has to have a disulfide bridge, it will not allow the fragmentation. Of course, incomplete peptides, trypsin digestion. So missed cleavages we have discussed. Incomplete, if it is a large peptide, more than 4 to 5 kDa, it will fragment poorly. We told it is much beyond the traditional range, and it will often be not identified without any specialized database. So suppose you are trying to look at a protein that only generates large fragments, you won't be able to detect genal. Is this helpful for you? Because you're the one that came up with the real problem in your research. Maybe you can go through these issues. Unassigned spectra, we just discussed. So approximately 60 to 70% of spectra remain unassigned. Can you imagine? That's why we have databases like PRIDE, where we put our spectra, and who knows, 5 years down the line, 10 years down the line, with advancements in computation, we can, and these 1,000 genome projects and 10,000 genome projects, we can know the human proteome much more better than what we know today. And maybe with better computing methodologies, we can again go back and reassess our spectra. So almost 60 to 70% of data is still lying in the database, not analyzed, just because spectra is usually not assigned to it. So what can we do? We can make customized data-based searches. We can customize a database. And of course, we need significant advancements in bioinformatics as well as databases. So these large genome projects that we are having, we can have, for each allele, we can have a different spectra. And then maybe we can just look for a specific allele. We don't want to look for the entire proteome. We just want to look for a specific set of alleles. What is the So I think his battery ended, and it was a signal that I should end my talk. So I would end my talk with a very nice paper. I think we will share it with you all. Like as I said, today evening when you go back home, or maybe tomorrow morning, you would find a drive link. So there are two things to do with the drive link. One is try to read all the papers that we share, and number two, I will ask all of you, you must have taken a lot of photographs. Do share the photographs if they are share-worthy, and specifically share if they are not share-worthy, so that we can have a meme fest in the group sometime later. Right? So that's all. I think that's my final lecture for this part of the day. I will now move for some admin work, and always remember, don't stop questioning. That's, I think, in today's date, when we rely so much on ChatGPT for almost everything, don't stop asking questions, because I think that's the most important thing, that's the most beautiful thing that a human does. Whatever we have achieved is because of our curiosity. Right? We are the only species who do that, because we have a very highly developed prefrontal cortex, makes us do things which a dog would find extremely unnecessary. Right? A dog does not have a planned meal. A street dog would roam around. Whenever he finds something, he will eat. If he doesn't find something, he won't eat. He will just go and sleep. You can't do that. We have long plans, right? All of you are here with the plan that, okay, I will learn something, then I will perhaps apply for a postdoc, perhaps I will use this in my research, perhaps when I am passing out my PG, I will apply to a proteomics lab, so it will help me. So we have long-term plans. Dogs don't do that. Again, all because of the prefrontal cortex. So please be curious enough. Don't stop questioning. And also, this is a hint that you can ask questions if you have any. Else, the tea has been waiting for you. You can join us for tea. And did I bore you? I mean, did I break your heart today? If it was a planned exercise, I think you already knew from the list that on day 5, we will know where proteomics fails. But I think after you learn that proteomics can do everything, you need to realize, just like any technique, it has its own pitfalls. And it's very important when we plan our experiments not to get over enthusiastic, and try to plan an experiment that is more pragmatic, that will lead to good results. Despite these issues, we have done considerable advancements in proteomics because of proper planning of experiments. Okay, so I think I will leave here. Okay, you have a question.
Sir, what would be your plan to learn the proteomics of a species which is not very much known in that field? Like it's only discovered in the field of proteomics.
You want to work on a species that nobody has worked on?
You have some papers, like 2-3 people have worked on this proteomics, but it is not in that directed way which I am working on. But I want to determine the proteomics of that species. Because in psychology, we identify new species every day.
Right. So has anybody done, like, if you don't go for the species, did anybody work on that specific class or subclass of? So you don't have to work on that specific algae. What was the work done on algal proteomics? Right. I would start from there. If I have to work on fungi, I would look what is the status of fungal proteomics. What are the databases they use? So there are a lot of things that you can use, like there are multiple softwares and that people use, which are actually helpful to try to extract data. But as I said, even with a known proteome, you just saw what are the challenges. There is almost 50 to 60% of unassigned spectra. You would lose out a lot of information. You can go for targeted approaches. Maybe you can, if you know specific peptides, you can create a list that, okay, you want to look at only 50 or 60 proteins, and you can run your experiment on a triple pod. Rather than doing a discovery approach, you can go for a targeted approach. You can only go for something that is already known, whose parameters are known, whose sequences are known. Because sometimes going for discovery at the level of proteome is difficult. Or you can go for the proteogenomics approach, where you can first start by sequencing the genome of your species, right? And do a 6-frame translation and create your own database. And then you do discovery data. Of course, working on non-standard models is difficult. So your thesis can be just establishing a proteomics pipeline to work on your specific species of algae. That itself will be a 2 to 3 year work, sufficient for a PhD. But now maybe somebody from your lab can take that pipeline that you have developed, and of course when I'm talking about a pipeline, I'm talking about a lot of things. Like when you go for a 6-frame translation, you see the number of proteins that you identify will incrementally go up. Then you have to validate those proteins using targeted methods. So I think I won't step into that mine if I had to do more work with it. Only proteomics, that itself is 4 to 5 years. I mean, 3 years is a very good experience. If it is something that you have a mass spec at your door that you can access daily, but even if it's a central facility, you would be fighting with 10 other labs for the same facility. And again, challenges when there are central facilities. Now they would run some sample, right? And few peptides stick. They are very sticky peptides. Once they run their sample, they have effectively compromised the column. So it happened in our lab as well. So even in the lab, there are multiple people working, right? Multiple groups working. So there was a specific contaminant that was coming up in one of our data sets. And we did not know why it came in some and it did not come in others. So what happens in a lab is we randomize the samples, right? We randomize the samples so that there is no bias. Because if you don't randomize, you will be effectively biasing your experimental data. So what we observed was this particular, actually 2 to 3 protein set would always come when person X's sample was lined up just before us. But it took 3 to 4 months to realize that. And by that time, we had already done like 30 or 40 runs. So what we did was we excluded it in our analysis. But again, that would be, that could have been avoided. So these things come up when you practically start doing stuff, start preparing samples, lots of things. I think I have tried to cover almost all bases. And the review article that I showed you is a very good article. It also shows the strengths and the weaknesses. I focused on the weaknesses because that was the theme of today's talk. Anyone else? Yeah.
So can we regenerate the norm cell that has been degenerated to
Yeah, you can do discovery proteomics. But you cannot regenerate per se, but you can understand a developing nerve cell. So nerve cells develop, right, from stem cells. So a stem cell that is differentiating into a nerve cell, you can try to understand its proteome dynamics. Right? Now you try to see what are the events that are leading to its full generation. Fine. Now you take a degenerating nerve, understand its proteome dynamics, and then you can do a systems level analysis and try to see what are the proteins that are getting turned on while it is generating and when it is degenerating. And based on that, you can build up a hypothesis that if you can turn on these specific proteins or these specific pathways, then you can think of something like regenerating a neuron that is already dead. So you have to go back in that way.
So, like, whenever we are to find targets or anything, so we just go for, say, in proteomics, we go for a protein. But of a single protein, we have different isoforms also.
Right.
In some studies, it has been reported that this isoform is significant in this kind of.
Yeah, that phosphorylations is for example.
Usually, in normal cases, we just go for the main form of protein. We don't look for isoforms. So, like, how we should deal with it if we are into discovery proteomics? So, should we go for like normal protein kind of thing, or how we should look for isoforms?
So you see, you cannot randomly look for an isoform, and because I just told you what are the challenges of randomly looking at something. One thing you can do is develop a hypothesis. Suppose you are working on a system that has lots of kinases, right? You're working on cell division, and we know there are cell division cycle kinases. Right? So, which means there will be a lot of phosphorylation events. And since there are lots of phosphorylation events, you can try to design a phosphoproteomics pipeline. Phosphoproteomics. So we did not go into deeper into that because perhaps that would be a different GIAN course. Where for 5 days we'll just talk about specialized, extremely specialized proteomics approaches, but then the target audience will become extremely specific to those who do proteomics at the level of PTMs. So if you do phosphoproteomics, then phosphoproteomics becomes a big challenge because it's not a big challenge. Then you have to start preparing your samples likewise. It starts as sample prep. Most of the things you would understand starts at sample prep. Because sample prep will significantly alter the kind of output you will get. So yes, randomly you can do that. But I'm not very sure about the reliability of the data. But you can set on max point, there are parameters where you can set to look for changes in the phosphoproteome or acetylations, but they are not very accurate. Because most of the changes that you would be seeing might also be changes that you induced while processing the peptides. I think I can take one last question because then we are getting late. Professor Graham's waiting. Andromeda.
In order to create the theoretical setup, we need to input protein sequences, right?
Right. Faster sequences.
So where we get the sequence? Because we do not know the entire protein profile of what are the species we are working. So in that case, if at all we are giving some input.
So Andromeda, the faster file that you can give can be anything. Right? The faster input can be, you can take, suppose you want to expand your search. So rather than going for uniprot, you can go for tremble, right? Translated in email. So that will significantly increase your hits. So the number of identified proteins will go up. But since we do not know whether these proteins actually exist, it becomes a challenge. You can sequence your own genome, you can do a whole exome sequencing, and from that genome you can do a 6-frame translation using a proteogenomics approach and make your own database. But every time you do that, the larger your faster file that you upload onto Andromeda, the larger the number of targeted theoretical spectra increases, and now the instrument will try to match first at the level of MS, then at the level of MSMS, and then give you the data. So theoretically yes, but again availability of time, availability of resources, all those will determine what best should you do. So you have to, so your name? Kishore. You are from? Manipal. The Manipal I think has a mass spec. Which one is that? I guess they have a fusion. So most of the central facilities that we have, they run thermal instruments. I think that way Professor Sanjeeva's lab is one of the first in India to start with the Bruker TIM stuff. So maybe in the next 4 or 5 years, now since Bruker has set foot in India, he would find more and more TIM stuff. So there are multiple things that you can do with mass spec. There is something very interesting that is mass spectrometry imaging. There you don't take samples in a solubilized form. You take a tissue section, right? Now imagine something as complicated as a brain. right? Number of cells, the way the cells are arranged, you have the hippocampal formation, if you have the prefrontal cortex, you have the cerebellum. Now, if I homogenize them all, I'm not getting the exact data, right? I'm getting a projection of the data, what it might look like, but not the exact data. So there you can do mass spec imaging. Mass spec imaging will give you this chemical map of which protein is present in which area of the brain. Also, there are other methods like thermal proteomics, chemical proteomics, so lots of things to do. I mean, you can explore a lot. I think this GIAN course is primarily to act as a primer. So we have added the PCR primers. Now if you have enough of stack polymerase and you are ready to go through those rigorous thermal cycles of heat and cold, then you would have a long amplicon. Okay, thank you. So please have your tea, and if you have not taken your attendance, do it. This is compulsory because if you don't have it, they will withhold your certificate. And after lunch, you have to take a test. If you're not taking the test, again, the certificate will be withheld. Anybody who wants to leave just after lunch due to train schedules? When are you planning to leave?
Yeah. Okay.
Thanks.
Right. Okay. So this next piece do you have some people in the front here, or are they at the back? Is somebody missing? No? Okay, so this session is about integrated multiomics and how we might think about strategies for using that. Anybody tell me what multiomics is? Any ideas?
Multiomics means like there are different biological studies, genomics, proteomics, transcriptomics, metabomics, everything. We study in a single study.
Do you think we have, or do we think we get a complete signature from multiomics? No, it's difficult to get a complete signature. Often it's sparse. You might have genomics and transcriptomics. You might have transcriptomics and proteomics. But because profiling is expensive and samples are precious, it's only more recently where people start to do multiomics. And people call multiomics, it's a catchphrase, it's a buzzword. And I've seen people call single-cell RNA-seq analysis multiomics, but it's not. So just have to be careful a bit about when people use this term and what it really means. Because multiomics should really represent characterization of a population across more than one of the transcriptional, translational pathway elements. So, having said that, multiomics is a useful data set, or a useful data to have, because one of the big issues around any biological profiling is heterogeneity. So, at the end of the day, first of all, every single cell in your body is different, and it will have a different molecular profile. So, single cell profiling is a useful thing to have. Having said that, single cell transcriptomics, single cell RNA-seq, you have to be careful of quality. So, for example, in single-cell RNA-seq, we might set out with 100,000 cells, or I use the word cells loosely, because maybe 20,000 of those cells will be dead. So the profile is not relevant for those. Maybe 10,000 of those cells will be fragments of cells, so we can't use those. So, and then, within those cells, we may have sparse or very incomplete transcriptomic profiling. So we might have only expression of only 2,000 genes. Now, if we want to do a very thorough study, we need to have a pretty complete transcriptome for those single cells, and that's challenging because they are very small volumes of RNA, and there's kind of a bit of an integration and an overlapping that goes on in single cell RNA-seq. But if we get it right, we can then have the potential to, say, look at a pathway model in fibroblasts, tumor-infiltrating lymphocytes, epithelial cells, stromal cells, and start to really get into systems biology, where we're looking at multiple cell types with RNA-seq data. One of the emerging areas is single-cell proteomics as well. And given that some mass spec, and one of the techniques that Professor Srivastava showed was a 4x proteomics, which essentially allows measurement of nanoliters and picoliters of protein. So you're starting to get to the sort of volumes of a protein you might have in a cell, certainly on the edge of it. So single cell proteomics is an emerging technology. It will open up all sorts of interesting avenues. The key thing, though, is it deals with heterogeneity. A tumour or a diseased tissue or blood is a heterogeneous sample. You can have multiple genetic variants, you can have multiple transcriptomic features, you can have multiple proteomic features. And looking across all of that starts to help untangle some of that heterogeneity. So what we're seeking to do here is to capture variations that are introduced by genes and their mutation. So do you know the difference between germline and somatic mutations? So cancer, you've got an extremely heterogeneous system because of various somatic mutations that are coming into that tumour because of errors in the chaotic environment, essentially. So again, understanding the mutational state of a cell can have an influence on how that cell behaves. I'm going to show an element in a little bit where we've looked at, it's not single cell data, but we've looked at the effect of p53 mutations on a quantification of the p53 pathway in breast cancer. So we can also get variation introduced by microRNAs, non-coding RNAs, long non-coding RNAs. Now these things are not necessarily coding proteins, but they can have influence on what is on transcription. So microRNAs modulate genes by blocking and stimulating the transcription of the gene. You also get methylation. So genes are suppressed or stimulated based upon the level of methylation. So methylation is silencing genes. So you can have expression of a gene, but unless you look at its methylation, that gene could have a strong signal, but it might be methylated. So we have to consider that level of the epigenetics. We also have copy number variation, so genes can have multiple copies. That sends more signal. We can have translation, so errors can creep in translation. Proteins may be created. We get protein expression. Then on top of that, we start to get PTMs, so post-translational modifications. So if we can encapsulate those, then we start to understand how the protein signal is being modulated and changed. And then last of all, we get metabolites. And the metabolome is important, and that's kind of the end product. In fact, I would say the proteins are the end product. The metabolome is a byproduct that's kind of tagged onto that because it's the proteins that are doing the functional work. The metabolites are a result of the protein's action. My view is that metabolites are much less diverse than proteins, and that occurring as a result of biological processes in the protein. We can also look now at spatial omics. So, have any of you heard of nanostring DSP? So nanostring DSP, you have a slice of tissue, you create regions of interest, and you get counts of sequence. I think last year it may have changed, it become even more. You've got measure essentially 18,000 genes on a region of interest. They're moving towards it being a deeper sequencing, even though where you measure the whole transcript. So you've got potentially transcript level information as well as gene level information for a region of interest. So nanostring have just been taken over and acquired by Bruker. So what did Bruker make? Sorry? In the context of this course, what did Bruker make? Mass spectrometers. So Bruker make mass spectrometers. Bruker are interested in spatial transcriptomics. So you're starting to get a potential towards spatial proteomics as well there. Because you have a bead-related approach which measures transcripts, gene products, at the mRNA level. But you could quite as easily have a bead product which measures proteins. So then you have spatial proteomics using a bead-based approach, which is like a protein array, and has the capabilities of a protein array to measure lots and lots of proteins with great sensitivity. So spatial is important. The next layer after spatial is temporal. So if you've got time series data, these things are all varying in time. Yeah. As I said at the beginning of this, consider systems biology or biological systems to be a concert where all sorts of things are playing together. Some things are influencing other things. Things are coming in temporally at the moment and leaving, becoming louder and quieter. And it's an interplay between all of these things in the state. And multiomics, as its higher goal, is seeking to try and measure all of that. So we've got single cell omics and we've got temporal variations. And then, of course, all of this is happening through a population of individuals. We're not just modelling one person. Every individual has their own unique history. They have a lifetime of accumulated mutations in some cases. They have a particular diet, they have a particular ethnicity. One of the statements made about proteomics is you could, by looking at blood or serum proteomics, you could probably work out what somebody had for breakfast, because it's such a dynamic state. It's and that is a positive in that it's really useful data, but it's a negative in that it's introducing lots of variations into the profiling. So what really I'm interested in, and I suppose it's where the subject is kind of heading, is systems biology. And systems biology is the mapping of things over space, over time, at an interaction level. To truly have systems biology, we need that multiomics data over space and time. So we could ask the question, what is the interplay between multiomic elements? How are they interacting across cellular populations, across time, and differentially between a disease state and a non-disease state? So in an individual, we could look at tissue from a healthy part of an organ versus a diseased part of an organ, assuming there's no field changes. So a field change is where the disease is causing a change to the molecular profile of the tissue surrounding it. So we have to be careful of field changes in this sort of study. But through untangling that, we can start to understand what drives disease, what makes it tick, what's the basis, what's the cause of disease. So what happens then at the network pathway level when there's interplay between molecular features? So, as I said, by looking at a map of the pathway, by modelling that, you can get a quantification of the pathway. You can quantify that pathway across multi-omics levels, but how do things change? If there's a particular mutation, how does that impact upon that pathway? If it's a different time of day, how does it impact upon that pathway? If you have different population groups, how does it impact upon that pathway? What's the effect of indels, insertion deletions in the genome on that pathway? All of these things potentially have to be considered or could be considered. Because if we understand the system and how the system changes, we understand the various colours, for want of a better word, of disease and how that spectrum of disease changes. So to illustrate this, there's a piece of work which I've done, I'm going to show you, which is modelling the p53 pathway in breast cancer by and quantifying that model, that interaction model, in different mutational states. So what impact does the mutational state have on the pathway? So before, we perhaps might have just looked at a pathway not considered a mutation. But in reality, if you've got a pathway that the core element of that pathway is mutated, it's not going to behave in the same way. So by looking at wild type p53, by looking at loss of function p53 and missense, also nonsense p53 and missense p53, we can see how does that impact across the pathway. How does it impact on its stability and what features are coming out. So there were components of the multi-omics data. I'll just zip through this. We've got the genome, we've got the transcriptome. Where are the limitations? Well, we've mapped the genome, but we haven't mapped it across the whole of human diversity. Now, there are programs developing, every country has its 100,000 genomes, but historically, genomes have been mapped for Caucasian people in the UK, US. India now has a 100,000 genomes program, which is developing. The Chinese have got a 100,000 genome project. More diversity is being mapped because there are different splice variants, different insertions, deletions that occur in different populations. We did some work on TB in China. A few years ago, we developed a signal based upon a South African and Caucasian population. And it turned out that one of our genes for a Han Chinese population had an insertion. So when we were doing PCR, we ended up getting 2 gene products for that gene insertion, which means that we were getting 2 signals instead of 1. So we had to resequence that gene for a Han Chinese population so that we could get a contiguous product, contiguous measurement. So the genetics is important. The transcriptome, big problem with the transcriptome is ontology. Now we need to do more and more functional gene studies. So many genes, if you look at their ontology, they're biased towards cancer, or even more so breast cancer and lung cancer. That's because that's where they've been studied the most, but the functionality isn't truly represented by the ontology. There are gaps in the ontology. There are functions which need to be fully defined. So this is one of the problems with a lot of databases, is the database is incomplete. We don't have a full ontological representation of everything. So be careful of ontologies, because, you know, one case in point, if we look at Panther ontologies, in most gene lists that we look at, we see Alzheimer's disease and we see Parkinson's disease represented, even if it's cancer, which is a concern. But the reason for that is that they basically lumped in so much functionality into Alzheimer's and Parkinson's because they don't really fully know how it's working. They're hedging their bets. So it's over-biased, the ontology, towards that disease state. It's not a real representation or unbiased representation. So ontologies are useful, but they are incomplete. The other thing is, with RNA sequencing, are all of you familiar with RNA sequencing workflow? Do you know the bioinformatic workflow to go from FASTQ files through to TPM values or RPKM values? Yeah. Well, when you look at the transcriptome, you can not only get gene level information, you can also get transcript level information. So that will give you the different transcript forms of the gene and the amount of signal coming from those. So some genes will have 15 transcripts in them. And across a population, you can have multiple transcriptional forms, which represent splice variants of that gene, essentially. And if you want to go even further, you can get exon level information for that gene as well, which gives you even more information. So we don't have to just look at the gene level, we can look at the transcript level. And in fact, I've got a project which I'm running at the minute where I'm looking at the relative contribution of multiple of the different transcriptional states to a gene and predicting that to understand what impact that those transcriptional states are having on a pathway. Because there's maybe different behaviors depending on the splice variant of the gene. So the spliceome is another level of complexity, and we don't really fully understand what that's doing. So that's another area of exploration. There's always more work to do. We can always go deeper, and this is the nature. In 1922, a group of physicists said, "We know everything there is to know. Science is dead." Then they discovered radiation. And perhaps it was before the 1920s, but there was a group of, I think they were either American or British physicists who were quite boisterously said and arrogantly said, "We know everything there is to know." But the world is such a complicated place, there is so much more to know, and we can go deeper and deeper. And that's, you are young, I'm less young. This is your future. There are an awful lot of things you could look at in the future, and there will be more things to look at as well. So, never think we know everything. The other thing, which is a gap in our knowledge, is there is a disconnect in multiomics between the message coming from the gene, mRNA, and protein expression. So, if you look at multiomic studies and look at a set of gene messages, looking at mRNA from RNA-seq, and then look at protein expression, there is a block of proteins and genes that turn up. About 60-70% both will turn up and be going in the same direction. There's another 20% where there's no connection whatsoever. So we find the gene product, the mRNA, from the mRNA, but we don't find the protein doing the same thing. And then there's a smaller slice, even more, where the gene product goes up, i.e. there's more transcriptional message, but protein expression goes down. And we don't know why that is. But it suggests that these things are not directly linked, but there are other things like methylation or influences of other genes and other proteins on that system that are changing that regulatory direction. So we've still got to disentangle that. The problem is if we use fold change as a measure of importance, we will not disentangle that because it's about correlation, it's not about magnitude of expression. So that's another challenge which we are yet to meet. The proteome is challenging. There are 14 orders of magnitude of expression of the protein in the human body, going from probably albumin on one level to proteins that are expressing at sub-picomolar levels. At the minute, we can probably measure 7, maybe 8 orders of magnitude with proteomics. With array-based methods, protein arrays, that might go up to 9. With some of the new mass spectrometry methods, that goes up to 9 as well. But we are missing things. We don't measure the whole proteome, so we have gaps there. That's something which will change over the future. Instrumentation will get better. So through building libraries, we can more accurately find these things, and if we know what we're looking for, we can more accurately measure them. So the coverage of the proteome will increase. As I said before, there are few studies where there's full data, where we have genomic, transcriptomic and proteomic data sets complete. There are some in TCGA, there were some lung cancer data sets where we have DNA sequencing, we have RNA sequencing, and we have proteomic data. The depth map cell lines, do you know what the depth map cell lines are? Okay, this is a freely available resource on the depth map. Basically, the Broad Institute has taken a whole load of standard cell lines, literally hundreds of them, and they have undertaken RNA-seq on those and profiled them, and they have undertaken proteomics on some of them. Now, why is this important? Well, the depth map cell lines are cell models that are frequently used to test if drugs are working or not. So the cancer cell lines, the MCF7 breast cancer cell line is an example. So with these depth map cell lines, because they've been profiled, people are treating them with a drug and then getting an efficacy measure, and then trying to look at how the gene expression or protein expression is influencing efficacy. But more than that, I think it's still part of the Broad Institute, they've done a project where they've taken around 350, 400 depth map cell lines and then exposed them to about 30, 40, maybe even 70 chemotherapeutic agents. So what they've got is they've got the response to chemotherapy, and they've got the expression profile prior to chemotherapy. So that data can be mined in order to understand what features are driving sensitivity and resistance to chemotherapy. So there's the potential to explore that and look for personalized medicine. The problem is the cell lines are not as representative of the tumor as real human tissue. So what we've been doing is modeling efficacy and then taking that efficacy model into TCGA data, which is blocks of tissue, and then predicting across a population what the spread of efficacy might be. So it's extrapolating from a cell line which is probably derived from one person's tumour up to a population of 1,000 individuals. So we can start to explore virtual clinical trials by that method. We can try drugs out by modelling them. So what do we do with this? If we've got a workflow where we have integrated data, how do we use it? Well, first of all, we gather the data. We need to see what sources of data there are out there. We need to look into repositories, GEOD, TCGA, Array Express. We then to so this is a standard workflow up until this point. If you remember, we can check for batch effects using something like Disney plots. We can normalize and scale data. Well, if you've got FASTQ files, then we can normalize that data using and align that data using something like Galaxy. So the SRA, which is the NCBI's RNA-seq or RNA-seq repository, you can download stuff straight into Galaxy and then run a Galaxy workflow on that, converting the FASTQ, running it through quality checks, trimming it, and then getting counts. I use Callisto, to be honest. The latest method is Callisto. It's a lot quicker than some of the previous methods like string tie and ball gown and top hat, and so it's string tie and sorry, mental block. But these methods, Callisto is a more efficient method and a more accurate method. It's been shown and quicker. So the next step then is we're starting to enrich features. So we have data from each one of these pools, and we're addressing the question if we've got the same data across different pools. So we can, if they've got the same pool of data, we can create a meta table, a meta data set, which has copy number variation, methylation, proteomic data, transcriptomic data, even metabolite data. And what we can do is mine that whole meta string, and as long as we know where the feature has come from, which pool, we can then say, right, well, this is the combination of best features that are stable that predict this thing. So we have effectively, we might have things where we've got methylation elements turning up, we've got transcripts, we've got proteins turning up as all important in the thing that's defining the disease. And we've done that as a group in Leeds University in the UK, where we've taken multi-omic data, copy number variation, expression, methylation, and we've effectively pulled features from across that workflow. Another study which we've done, which I'll mention here as well, is there are publicly available data sets for Crohn's, for ulcerative colitis, as a part of a broader group of IBD. In that study, they've got proteomic data, they've got transcriptomic data, and they've got metagenomics and metatranscriptomics data from fecal samples. So they've got the gut microbiome there as well, which means that you can start to understand the interplay between the gut microbiome and the transcriptome of the host. So that's another level of dimensionality, but gut microbiome plays an important role. The point is, if we're doing this integrated analysis, there is no bias introduced. We're considering everything on an even keel. So once we have our enriched features, we can then identify the interplay between these things. So what's the interplay between a gene, methylation, and proteomics? And I said we don't really know how proteins and transcripts link. Well, this allows the potential for us to see how things are interacting that define the expression of a protein. Is it a methylation event? Is it a microRNA event? Are these things coming together? Is there agency from a third or fourth gene that's regulating the production of a protein? So we can start to dig into that. If we wanted to be really detailed, we could take a protein as a target and look across all the multi-omic data sets and see which things were influencing just that protein level. And that would start to find associations that might be driving the expression of a protein. So many experiments, only so little time. What we can also do, because we have genomic information, is we can look at mutational states of genes. So we can add another layer of complexity by rerunning this whole process for different mutational states of the gene. So how is this big interplay happening here for the wild type, and how does the mutation of a gene perturb all of that? Is there compensation? Do other proteins come to the fore? Do other genes come to the fore? Is there an attempt by the system to methylate a mutated gene or a set of genes resulting from that mutation? A set of transcripts resulting. How is this working together? You know, this is a very complex system, but we have to be a bit creative and think, how can we interrogate this? So that's the workflow where we have integrated data. Just bear with me, I just want to see if we... Oh yeah, that's good. How do we have... What workflow do we create for single cell data? Well, we gather the data, normalize scaling, batch effects. In single cell data, we have to remove dead cells, senescent cells, cell fragments, artifacts, and cell nets with low numbers of expressed genes. So we're kicking out the rubbish. There's huge amounts of debris in any cellular model. If you've ever looked at flow cytometry data, there's so many fragments, and it makes a big chunk of the spectral distribution. So we've got ways of assessing the profile of a single cell and eliminating it if it's got some problem with it. Now, what we then do is we've got all this mixture of cells coming together, and we have to determine what a single entity is. So the best available technique at the moment is CyberSort. That used to be free, they've just started charging, which is a real pain in the backside. But with CyberSort, you can say what type of cell it is based upon, they kind of use multi-dimensional scaling to derive this, but then histological evaluation of the cells found. But then, effectively, they've got a set of algorithms that look at transcriptomic markers to define what type of cell a given cell is. So it might identify it as an immune cell, it might identify it as a specific T-regulatory cell, it might identify it as an epithelial cell, as a mesenchymal cell, as a stem cell. But CyberSort is there to try and identify what type of cell it is. Anybody think there might be a problem with that? What could the problem with that be? Is CyberSort right, do you think? Any ideas? Be brave. There's no right or wrong answer. Is CyberSort right?
What parameters does it differentiate?
Exactly. It's a model. And okay, if everybody uses it, it becomes a very strong model, but it is still a model. Where does it fall down? When does CyberSort get it wrong? And again, this is a key point here. We always think about the workflow you have and whether that workflow is right or wrong. What is the error introduced by using CyberSort? Now, if you use CyberSort to predict cells, in order to look at the profile that defines a cell type, you get a self-fulfilling prophecy, because if you use CyberSort to predict, say, Tregs, and then look for the molecular profile of Tregs, well, those cells have been defined by CyberSort, they will always be Tregs. And the markers you find will always be Treg markers. So you end up with this reinforcement of an idea. So you have to be careful with any method and consider what its limitations are, and clearly communicate the model you've used. In the next 5 years, somebody else might produce something 10 times better than CyberSort. Does that mean that's all results previous to that are wrong? Now, be cautious. There is no right or wrong answer. There are merely ways of looking at a very big world through very tiny windows. That's the problem. Science is like taking a tiny peek at something that's the size of the universe, but through a window of 1 millimeter diameter. So be mindful, be cautious of what is happening in a data set. Yeah.
Can you explain how CyberSort actually works? Like what parameters does it take?
It's a classification model. So basically, they've looked at regions of a multidimensional scaling or Tisney plot from a mixture of cells. And then they've defined regions and then looked at the genes that are differentially expressed from other regions and said, okay, well, this is a mixed cell population. This is a population of immune cells. What have we got in there that's different from every other cell? And then they'll dig into the immune cells and say, well, what types of immune cell do we have here? But it is, I've oversimplified it, but they've done that extensively across multiple single-cell RNA-seq samples. They've enriched for certain cell types and then run it. So they're not taking every cell. They might, for example, take an immune cell cohort that they've separated out for various means. Then they'll profile that. But ultimately, they're looking for effectively determinants of a cell type and building that into a classifier model. That gets better all the time, but it's still a model, and every model, as I said the other day, has its errors. Is it a paper plane, or is it a full-size model of a Spitfire? That's the question you have to ask. So,
is it like in the manually way, like I've done single cell analysis. So what we do is we do clustering, and from the literature mining, we retrieve all the markers or cell type identifiers which are broadly categorized, like B cell, T cell, or macrophage, and then we extract those specific clusters separately, and then we recluster them to look for the subtypes.
Yeah, that's exactly. You can do that. That's a good way of doing it.
That is it's a good way instead of using CyberSort.
Well, you can use the literature. CyberSort probably uses literature to some extent as well. But yeah, as long as you've got a rational approach that you can state and be aware of what the limitations of it might be, then that's perfectly valid. Yeah. The challenge is you're looking at something quite complicated and vastly oversimplifying it. And so, for example, with clustering, what form of clustering do you use? Because if you use hierarchical clustering on a whole transcriptome, k-means, yeah, that's what I would use, yeah. So if you're using k-means clustering, that's good, because that deals with the high dimensionality. If you're using hierarchical clustering, there's as much distance between the individuals on a branch of a cluster as there are, so you get these these massive differences between individuals, but very small differences between the cluster. So the inter-individual distances are huge, the inter-cluster distance differences are really small. So be careful of using hierarchical clustering on large-scale omics data. So yeah, but that k-means is a good way of doing it. So it is a mixture of clustering, a mixture of evaluation, marker definition, and CyberSort have done that, but you can do that manually. If CyberSort is a quicker way, because as I'm sure you're aware, going through the whole clustering business and defining the markers for types is quite laborious. Yeah. But it is an accepted method, CyberSort. So how can we use single cell work? Well, if we use that concept of a digital twin, remember I showed that yesterday, and we're quantifying a pathway, what we can do is say, right, let's build a model of this pathway for mesenchymal cells, for epithelial cells, for stromal cells, for cancer cells, for stem cells. And how do those models differ? So, as an example, there's a piece of work I did for a large pharma company. And they were interested in autoimmune disease. We were in the final stages of writing this up as a paper with them, so I'm not revealing any confidences. So they were interested in this autoimmune condition. They'd got lots of blood data, they'd got lots of tissue data. So they had a hypothesis that there was a particular pathway, or two particular pathways, that were of interest. So they wanted to validate that hypothesis. So what we did was, the first thing we did was look at disease versus healthy for blood and tissue. So we ended up having an enriched set of markers for blood, for the disease, and an enriched set of markers for disease versus healthy. What we then did is use the pathway method that I showed you yesterday for both of those pathways, and enriched around the pathway for disease, blood, and disease tissue. So this gives us a list of features for phenotype of disease and a list of features for the pathway. So in one pathway we found we had 86% overlap with phenotype, so that's good, that pathway is valid. In another pathway we had 6% overlap. So they were surprised by this because they thought both pathways were relevant. So we'd validated that one pathway was there in bulk tissue and bulk blood. Where things got interesting is we then repeated that analysis for single cell of tissue, and we were looking at we looked at fibroblasts, we looked at infiltrating lymphocytes and epithelial cells. We then enriched the phenotype and enriched the pathway, and what we found was the pathway, which only had 6% coverage in bulk tissue, when we looked at infiltrating lymphocytes, we had 90% coverage. So the thing that was missing in bulk, when we went to specific tissue types, became apparent. So you can see how we can reveal mechanisms and validate mechanisms by looking at different types of data and seeing which context works and which one doesn't. So we then validated that pathway, but it only validates for one cell type. It doesn't validate for all the other cell types. So what was happening in bulk is that signal was being diluted. But when we look at single tumor infiltrating lymphocytes, it was strong and it was there. So that's an important thing to consider. We can look at single cell data versus bulk data and cross compare the two. So we can run network inference on the different cell types, and we can, if we have a pool of cell types, let's say we have stromal, we have epithelial, we have tumor, we have infiltrating lymphocytes. If we take the signal, let's say we've got transcripts, and we label the transcript name with where the cell came from, and then we pool that into one big data set, we can start to enrich around that and see, do markers from different tissue types in this mixture of tissue talk to one another? What is the strength of interaction between a T cell expression of IL-10 and a stromal expression of EGFR? That's a simple example. But by labeling with position, we can start to look at tissue crosstalk and to see whether there is crosstalk between cell types. That's of importance. So that's another layer that we can build into this if we want to. And we can notice the crosstalk between these cell types, and how is it differential between a mutant and a wild type state of a gene. So, you can start to see, this gets complicated, there's layers to it, but we can try and disentangle those layers. Any questions? Hopefully you can get a flavour for how all this multi-omics stuff that fits together, how we can use it to probe and interrogate data and understand what's going on. Is it clear for you so far? Are you okay? Yes? No? Okay. The next thing we can consider is spatial omics. So nanostring. So spatial omics is also being, does anybody here know about 10x single cell sequencing? So 10x have a new method called Infinium, and 10x Infinium is looking at single cell, but it also tracks position in the tissue and is looking at spatial single cell. Just another layer. It goes beyond what nanostring does. But we can do the same sort of thing. But here we're looking at regions of interest. And then basically we label the gene name with region of interest. So can we look at the crosstalk between a set of tumour cells, or between a tumour region and a stroma region? Much in the same way as single cell data, by labeling with region of interest, we can start to explore the interaction between these things. If we don't have integrated data, we have a bit more of a problem. Here, what we have to do is we have to enrich for the data sets separately. So we would enrich separately for transcriptomic, proteomic, methylation, metabolomic. So that gives us a pool of things for each one of those compartments, probably from a different population, because the same experiment hasn't been done on the same cases. But what we can do here is start to look for the co-occurrence of things. So does something crop up in the gene at the transcript level, and also crop up in the methylation level, and also crop up in the proteomic level? But we can't really say these are competing with one another, because the answer is coming from a different data set. So it's harder to do integration unless we've got a contiguous data set. So contiguous is where we have it all joined up. It's the same thing for, same set of cases for genomic, transcriptomic, proteomic, methylation, metabolomic. But we can look for commonalities and run the network inference on the commonalities. So, one case study, we did this some time ago. This is I think it's a European Journal of Cancer paper. We did this 2012 actually. And what we were doing is looking at array data, and for the same cases they'd got microRNA arrays and cDNA arrays. So they had integrated data, they had mirs, they had transcripts. Of course, now with deep sequencing, we have the mirs, we have the transcripts anyway. So they're all automatically there, so we don't have to worry about integration. So here we were looking at nodal status and estrogen receptor status in breast cancer. And we'd enriched for mirs and cDNAs. And then we ran network inference. And what we found was that certain mirs, we could see how the mirs were influencing the expression of genes, and how that was different between ER positive, ER negative, luminal A, non-luminal A. So we could work out what the drivers of that system were, but how those drivers were being modulated by microRNAs. And that's important because they're playing a key role in the whole system. Second case study, and this goes in a bit more depth, is what is the impact of the TP53 mutation on the TP53 pathway. So here we got breast cancer data. This is unpublished. We got TPM values, and for the breast cancer data we knew whether they were wild type, missense or nonsense. We knew the specific mutational points, but we then translated to those where they were known commonly as missense or nonsense. So we used the pathway miner bit, where we look at the addition to the pathway to kind of explore what was impacting on that pathway. So, but you know how I showed you we could mine a pathway? Well, we were mining, in this case, every single gene in the P53 pathway and looking at what interacted with it stably. The key thing is what interacted with a gene of the P53 pathway stably, because some things, if they're not stable, they won't be interacting. Now there's a lot of data here. I'm only going to show the tip of the iceberg. But what we were able to do with this is, because we had a map of what was interacting for every gene of the P53 pathway, we could see firstly how many stable features there were interacting with a gene. We could see what interacted with the gene, and we could see whether the gene for an unseen cohort predicted itself in the top 500. That's important because a gene, if it's stable across a population, you build a model to see how it predicts itself on training data, but that same function should carry on into unseen test data. So by using the performance on an unseen data set, we could see how stable it was across the population. And in some cases, as you'll see, the gene that was the output of the neural network model didn't even predict itself in the unseen cohort. What that means, it's highly unstable. There's message coming out of that gene, but it's complete nonsense. So we're able to effectively test the stability of a gene's interactors, and we can also test that stability in a healthy state as well. I'll illustrate what I mean. So this is the gene list. Here, instead of using the KEGG data set, we used the Hallmark. There's a few more genes in the Hallmark list. So what did we see? Well, in the wild type, there was a strong signal coming from P53. We saw 402 out of 500 things interact. So we're looking at the top 500, we're sorting based on the unseen performance, and then we're repeating that in 20 different cross-validations. And then each one we look at the top 500. So if something comes up, if it came up 500 out of 500 times, that would mean across the 20 repeats would have perfect consistency. Everything is coming up stably. If something comes up less than 500 times, then there's minor instabilities. Here we had things coming up 402 out of 500 times, which is pretty stable. The things that were coming up were pretty consistent and stable. Does, in this case, P53 predict itself across multiple data sets? So remember, in that list of stable interactors, ideally we should see P53. It should be predicting itself. And it did. And then if we looked across the whole pathway in the wild type form, how many additional new features were there? Well, in this case, if we look at all that stability, there were 2,652 new stable features coming into that broader pathway environment. So again, that suggests that it's mapping a very comprehensive and strong signal. So that was what happened for wild type. If we looked at missense, so this is a disruption of the signal. it's noisy. In missense, how many P53 interactors did we have? 1. So we had 1 interactor out of 500, and that wasn't P53. So we found something which was probably a false discovery. P53 does not predict itself. So in the missense setting, P53 was highly chaotic. It's producing loads and loads of signal, but the signal's complete rubbish. So P53, in this case, is not doing its job. It's not predicting itself. It's losing information. If we look at the number of genes in the stable features that we found, we only found 90. Those 90 are probably compensatory mechanisms dealing with the loss of any signal, or dealing with the aberrant signal coming from P53. So that's the mutated missense state. We also had a non-sense state. Here is kind of a halfway house. We saw 245 things predicting P53, so it is something's happening here. It might just be that it's a low level of expression. It's not completely deleted. It's effectively not much signal coming from it. P53 did predict itself, and we had 393 genes stable interactors. So we've got a weak signal here. It's not as clear as the wild type. It's less stable, but it's less stable because there's less signal. So with this, we were able to effectively say we ran all these lists through network inference, and we were able to say what perturbations arose in the pathway using a hypothesis-free, evidential-based approach, and how different mutational states perturb that. Now that's important because if you want to find a treatment, if you know you've got a patient who's wild-type P53, then you can find treatments for them that will help deal with their cancer in the context of wild-type P53. If you've got a missense P53, that creates a different state, and you're dealing with the compensation system where P53 signal has been perturbed, messed up. So you treat that patient in a different way. This suggests where you treat them. And then the nonsense setting, again, you have a different setting where you would treat those differently. Now there was a small degree of overlap between these three. There were common features, but there were specific features to each one of these states as well. So we have the 2652, but there were not many of those 90 that overlapped, and there were a few of the 393 that overlapped as well. So we were able to see where the overlaps were, where the commonalities were, and where the differences were. So that's ending this lecture. Any questions? I've kind of taken you to the tip of Everest in terms of the amount of stuff we can do with omics data, so it's probably a lot to take in, but what I would say is read the notes, contemplate it, and see where you get to. But any questions now? No? Okay, what's the time? Right, I think we can probably go to the last lecture now, which is not a huge one, and that will mean we've done my lectures before lunchtime, and then we can start getting into other things in the afternoon. Right. my view of what the future might look like for medicine. It's science fiction, it's not science fantasy. If it's science fantasy, it would not be real. If it's science fiction, it's an extrapolation of what we have today to what might be developed in the future. So it's where medicine might be going, where we might see some things happening, and how much. But it's done from a proteomics, multiomics, informatics, AI perspective. So let's see what we think. And you're the people who are going to have to develop this, by the way. No pressure. So, it's a statement by Sir William Osler, who was quite advanced in his time. This is 1892. And he said, if it were not for the great variability amongst individuals, medicine might be considered a science and not an art. Any medics in the room? I have had a few people throw shoes at me for this statement and get quite upset. But what its meaning is, medicine, as it was practiced then, and we've got a long way since then, is based upon observation of symptoms. It's not based upon the molecular characterization of a disease. They just didn't have the technology. With the post-genomic era and with the advances in various multi-omic techniques, we now have the potential to define a molecular symptomology of disease. So we can look at molecular profiles and understand those molecular profiles in order to determine the causality, but also the heterogeneity of disease. So, medicine is and was based upon observation. And the descriptors, so the nomenclature, for want of a better word, of medicine, is based upon the symptoms, the location, the body part. And by the location, I mean the location in the world, not the location in the body. The body part, the person who discovered them, the age in which they occur, the causative organism, which is probably about as advanced as they got then. So medicine, there's no standard way of defining these things then. So do you know about ICD codes? ICD-10. So what ICD-10 codes are an attempt at a standardized nomenclature. It's a code that represents a disease type and a disease subtype. So it's a standardized notation for a disease. So at least that is starting to come together, at least from the disease perspective. It doesn't use this loose language that they have. So it's a standardized code. So that will be important because people, you can use an ICD-10 code, and I think they have also an ICD-14, which digs deeper, but this code defines a particular disease type and subtype. So, but how were they characterized? Well, hypertension's first example, that's a symptom. But people call that as if it's a disease. And actually it's a condition. Japanese encephalitis. Does it mean it only occurs in Japan? No. It was probably prevalent at some point in Japan. But why does that make it any clearer? Whether it's in Japan, UK, America, Brazil, whatever. It's an irrelevance. Breast cancer, well, that occurs in the breast. Alzheimer's disease. Why do we name Alzheimer's Alzheimer's? Does it occur in the Alzheimer? No. Big grammatical clue. It's got a capital A and it's got an apostrophe. It was developed or discovered by Alois Alzheimer in Germany. So Alzheimer's disease was discovered by Alzheimer. Senile dementia, well, it's sort of an age group. And amoebic dysentery, well, it's caused by amoebas. But you can see how there's no standardization here, it's all over the place. So the taxonomy is diverse, doesn't define the disease, and it kind of is a bit of nonsense, really. So this makes it hard to treat. We have a numerical nomenclature in the ICD-10 codes, but really we should also be having a molecular nomenclature for a disease state. Some point in the future we will do, I'm sure. Now, we can start to get to that in the post-genomic era, because now we can start to look at the potential for the characterization of diseases at the molecular level. We can measure the disease in a molecular basis. There's problems because there's a lot of information. That's the whole basis of this program to some extent. Diseases are diverse and we have to search through lots of information. Fine, we've discovered that and discussed that. And we should ideally have a small set of features that define a disease from others. So what sort of personalised medicine is there? And this is how medicine may go, and it is going this way. So first of all, we have diagnostics. The diagnostic defines somebody with the disease from somebody who doesn't have the disease. So for example, prostate cancer versus BPH, benign prostatic hyperplasia. We also have prognostics. So a prognostic tells us how somebody's going to do. And in a basic form, we might say, what is the probability that a breast cancer patient will undergo metastasis before or after 5 years? More specifically, we can look at how long will it be before. So one example might be, how long will it be before this disease progresses? We predict with our algorithm that it will progress at 11 months. That's a more accurate prognostic. Just one point around accurate prognostics, you have to be careful because yes, you want to identify the risks to somebody, but it's like saying to somebody, you are going to die on the 12th of December 2019 or 2029. How does somebody deal with that information and process that information? So you have to be careful about how you present it. Usually it's low risk, medium risk, high risk. You don't tell somebody the day they're going to die, it doesn't go down well. So there's an ethical issue there. And the third type is predictive medicine. And this is where personalised medicine starts to come in. So which individual will respond to a therapy? What's the probability that this individual who has CML will respond to imatinib? It becomes a tool for prescribing, a tool for treatment. So that's the key next step, if you like. Imagine a situation where we fully understand, with companion diagnostics, this is what they're called, the best treatment to give to a patient to maximise their response and minimise their side effects. Now, with the data we have, with the methods we have, and we need to have the buy-in from pharma to some extent, but with those things coming together, we have the potential to start to look for personalised medicine and to give patients the best possible treatment. Now, in some cases that exists. So, for example, if in breast cancer a patient is ER positive, we know that we can give them tamoxifen. If a breast cancer patient is HER2 positive, we can give them Herceptin. And then if they're triple negative, we try the basic various cycles of chemotherapy. But to give a case in point, those simple predictive response tests have increased the likelihood of survival to 10 years for breast cancer patients by about 80% over the last 10 years. So if you know how to treat the different heterogeneities of a disease, you can massively increase survival and de-risk that for populations. So that treatment and personalised treatment is already yielding benefits, but it can go further. So where are we going with data generation? Well, in all likelihood, we will see an increase in coverage of the proteome and metabolome, so we're getting more data, more depth of data, more accurate data. We'll get an increased depth of sequence of the transcriptome, although that's not got that much further to go. We'll get more accuracy with reduced sample volumes. So we'd be talking picolitres or femtolitres of blood, getting a diagnosis. Non-invasive tests, so there are already technologies that, based on putting a patch on your arm, you can draw interstitial fluid in a non-needle based approach. So you could have patches which are taking samples all the time. Yeah. The other thing is there's always a trend of miniaturization in technology. You know, when I first had a mobile phone, it was about that big. Well, it's quite big now, but they packed more data in now, but phones went quite small for a while. First computer I ever saw took up a room the size of this room, with a little terminal that sat at the front of it. Technology is miniaturizing. So if technology is miniaturizing, can we have a handheld mass spectrometer? Can we have handheld real-time sequencing? If you've seen an iron torrent, by the way, iron torrent sequencing, little cassette about so big. Things are getting smaller. Technology is becoming more compact. Could we develop a real-time iron torrent sequencer or real-time sequencer or a real-time mass spectrometer that gathered data on my blood proteome continuously? Cost will come down, which means we end up with more data, we end up with more frequency of measurements. At the end point of this, can we have point of care diagnostics, so a device that does mass spectrometry, let's say, wearable proteomic devices, or wearable transcriptomic measurement? Now, if you're measuring the whole proteome and the whole transcriptome, then the data you're generating doesn't only answer one question. Imagine a situation where you've got a wearable device measuring your blood proteome, detecting all sorts of issues and risks. You have the potential to monitor risk of 50 different diseases in real time on a daily basis. Okay, your markers are up for this, the risk of this disease. Go and see your clinician. They will find out the best treatment that you have, catch that early, or even give you lifestyle changes to prevent that thing even occurring. So we have the potential to get to this point. In the interim, it's early detection of disease. There are cancer programs now which are looking at people who are being detected pre-symptomatically. Or, if we're gathering data about our molecular profile and our lifestyle, given the pool of thousands of other or millions of other individuals, can we make suggestions about lifestyle that will prevent us getting the disease in the first place? This is starting to sound a bit like Ayurveda, by the way. So prevention is even more powerful than cure. In trends in diagnostics, well, we have early stage diagnostics, but are largely knowledge is hypothesis based. Often there's a high attrition rate for developed tests. These tests fail quite often. With increased data, we get greater population coverage, we get better temporal and spatial coverage. There's a huge opportunity for diagnostic discovery. This will rise, there'll be a rise in the increase of multifaceted diagnostics. So an algorithm that looks at your whole proteome, as I said, diagnoses the risk of 50 particular conditions. Currently, it's one marker for one state. So P53, HER2, ER, single marker measurements, giving one state. Sometimes it's maybe a small number, 1 or 2 or 3. But if we move to a whole proteome approach, we can have multiple markers, multiple states. Now, if you look at most cars now, people plug in a diagnostic device. It looks at all possible problematic states of that car and says whether there's a problem. With a particular sample from a human, can we look at all possible disease states and problematic states that might be going on for that human at that point in time, based upon a complete proteomic or transcriptomic profile. Blood is kind of the universal diagnostic fluid because it encounters the whole body. So if you've got blood markers, it's relatively non-invasive and can detect things and check for things. So there's a move to multi-markers, multiple states, a move to prognostic models, early detection of disease and personalised medicine, and we can potentially couple therapeutic development with response to therapy. So this is starting to happen. Drug companies, by the FDA, are required to put in companion diagnostics. That is kind of loosely adhered to at the minute, but if there's the right data, you could actually couple a clinical trial with response predictors in order to be able to develop a personalised or companion diagnostic for that treatment. Trends in AI and data analysis techniques, where's that going? Well, we are continually getting higher performance of computing. We're getting greater explainability into that. We've got kind of a synthesis of LLMs coupled with evidential approaches, so people are developing LLMs, validating them with real-world data. We've got better algorithms for integration of multiple data sets, multi-omic data sets. We've got development of digital twin models. We've got development of in silico drug response models, virtual patients and virtual clinical trials. So based upon data, we can simulate the potential of a drug to change a system. I mentioned to you before, DepMap, the data there, that will go further. Trends in treatment and drug discovery, well, we're going to have more integrated pipelines of discovery. We can model currently biological potential. We will go further and look at the ADME DMPK. So this is about the metabolization of drugs, about the efficacy of drugs, about where the drug goes in the body, about the degradation of drugs, the half-life, whether it binds to fats. All of that can be modelled ultimately. For early diagnosis, we can identify determinants of disease. We can look at preventative treatment. And there's a big move towards ageing lately. So we can look at age-related diseases and start to find cures for age-related diseases. And if we know systems are dysregulating, there are mutational forms of genes, can we correct those with CRISPR-Cas? So can we treat the issues arising from accumulated mutational states over life with CRISPR-Cas, give you back the genome of a 2-year-old? Because aging results in, of course, it occurs because of rising mutations. It occurs because of telomere length dropping and a whole load of other genetic changes that are occurring in response to your environment. So we can see and expect an increase in gene therapies and also an increase in personalised medicine. An emerging area is synthetic biology. So people are already building virtual cells based upon molecular organisation and engineering. So with these we can build better models of disease. One of the big challenges for many diseases, particularly neurological diseases, is getting good models of diseased neurological tissues. We're starting to see some of that. People are starting to create much more robust models for neurology. But if we can get good representative models at the cellular level or at the virtual, at a cultured organ level, we can create an artificial cell-based heart which we can then use for treatment. It moves away from animal models which are not representative, but moves towards something which is more representative of the human state. One of the things that's also potentially emerging is the use of stem cells to grow organs on a chip or regrow cultured organs in a vat. So we could grow somebody a replacement heart, liver, kidney, so on. Based upon stem cells, hematopoietic stem cells which have been induced with induced pluripotency from a sample. So you can take blood cells, induce iPSCs, differentiate those iPSCs to tissues, use those to create an organ replacement. We will also develop in silico, molecular level, tissue level and organ level systems models and models of the interplay. So you can imagine through having digital twin of a system, we can join all this together to create a molecular virtual human. Which means you can start to do things like create this temporal model. What happens if I change this gene? Suddenly this gene mutation arises in this tissue. How will that effect cascade through the whole organism? There's potential to do that. The guys who discovered AlphaFold are currently building a virtual cell model based upon all the pathways in a cell and using that to create a dynamic model of a cell. There are limitations. Which cell are they choosing? How do they validate that as well? But there is, again, move towards a virtual digital twin of a cell. I was at a society in London, and this statement came from the guy who won the Nobel Prize for AlphaFold. He was presenting that at that meeting. So we could end up with whole individual digital twins. I give the system your genome, and it will consider a version of you which is a digital twin of you, which is generated through AI, but also through systems level models. So we can then look at experimental treatments on the digital twin of you. How will you respond to this drug? Can we, run a scenario where we give you a treatment without you actually even ever having the treatment to see whether it cures a problem? Does it cause problems down the line? Are you going to have an adverse reaction to this drug if we give it to you, based upon you as a digital twin? So that's quite radical, but it's kind of a view from my perspective. I'd be happy to answer any questions, but feel free if you've got a radical disagreement with me for any of this, please. I'm quite happy to hear what you have to say. Have I stunned you into silence? Is that what it is? Any questions? Yeah, well, that's this whole hypothetical, and that's fine.
At our level, like not with so much advanced techniques so at what level can you go to design a, not cellular level, but at least a few combinations of pathway interactions that go model? How we can do?
Yeah, so the method I showed you in Weka, if you wanted to look at how things are interacting and how stable they are, the method I showed you in Weka, where you're mining across a whole load of things and getting weights out of those things, so you're getting enrichment and then you're getting weights out of the Weka model, you could use that to start building an interaction model of a system.
Suppose we are giving a transcriptome data, then we will be giving it in raw form or after validation?
So it doesn't matter how you give it. Say you've got a FASTQ. That's the raw form. If you know which version of the genome library it was run under, then you can run that through Galaxy to get gene level information, transcript level information as TPM values. So if you go to the raw form, you can use that. It usually takes about a day on Galaxy to run that. So you've then got the normalized gene length corrected data for that human. Once you have that, then let's say you've got a particular hypothesis or question you're exploring, then you can mine that to enrich the features associated with that question. And then maybe there's a different state, maybe there's a mutational state, then you can mine that to see the features associated with that question. And you're getting gene lists each time. Then what you want to do maybe is look at how those genes predict one another.
Maybe like small molecules sensitivity.
Yes. So you could, in that enriched list, maybe it's 500 genes, maybe it's 100 genes, running that through Gnome AI, drug Gnome AI, running that through Campbell, running that through Reactome. All of these from this list, then you've got all sorts of other options to explore where those genes are, what they're doing, how you can interact with them, how you can drug them. If you want to create siRNA against them or CRISPR-Cas against them, you can find the mechanisms of that very easily in gene cards. So you've got it's like a tree. The first branch of that tree is which genes are associated with this question. And then you've got all sorts of different routes you can go down to explore what they're doing, what the context of them is, which pathways are they in, which drugs target them, and so on. And that's really what all this is about. It's about workflows going through from data. So there's nothing really to stop you doing anything. If you've got data, or you've not got data, you go get public data, you can start to explore all of this. You don't need a lab, all you need is a computer. Yes, you need a lab to generate aspect data, and you need a lab to do RNA-seq. But in the absence of those, there's a lot of public data out there which you can try things out on. So one thing I would say to you is go out, look for data, have a play with that data, try some of these things out, and start exploring, start experimenting. At the end of the day, you're running experiments, you're just not running them in a test tube, you're running them on a computer. And with this course, you should have the components necessary to do that. But if you get stuck, email me. If there's a question you don't understand, how do I make this step? I've got these things, where do I get to next? Let me know. Any other questions? So you're going to now change your proteome by having lunch. And please ask me questions if you don't want to ask questions in front of everybody else. Thank you. to the back of the room. So those at the back are going to have a harder job than those at the front. So if there's any better reason to move towards the front of the room, I can't think of one.
So the questions will become incrementally tough when we go band, because most of the common stuff the guys sitting ahead in the front row will answer. So to the webcasting team, you can just pan your camera when it goes back. And I think we still have, the second row is still empty. So Shivani Chandra and company.
3 high-value seats here.
Yeah, all of you, even the volunteers can come in, and it's not necessary that you would learn something from proteomics or AI and machine learning. Anything that this 5 days, almost we are at the end of the fag end of the 5th day. So of course most of you would have started off from your home. Professor Graham started off from UK on 20th or 21st? 20th. So for him is like the 10th day.
I didn't come by horse. I did come on a plane. It didn't take that long, but I stopped off in ICB. But
so he's also a visiting professor at IIT Bombay, and he also visited. Is the webcasting on? Yeah. So Professor Graham also visited as a keynote lecturer to deliver the keynote in Proteomics Society of India. It's a nice society if you want to meet more people doing working on proteomics.
The next annual meeting is in Mangalore. So if you want some time by the seaside on a conference, the PSI meeting, the next meeting next year is in Mangalore.
So yeah, you can come ahead. We have a space here.
This one here.
Yeah, Reshma, you can be comfortable. You can use that chair. Okay, so do we have one more mic? Is it working? Yes. Hello. No. Hello. Yeah, it's working. So I hope it doesn't start ringing. Hello. Yes, pretty. You can use it from here. It's pretty. The gain is pretty high.
Yeah.
But I think I will pass this on. So you can start from the lady there.
So yeah, name one thing you've learned.
You can tell your name and then stand up. You can pan. लेकिन वेबकास्टिंग कर पाओगे? I think it's fine. You can just pan. Put it on a wide angle. You can pan it, and you can face the camera, right? Right. That will be.
So now you can give lectures.
Okay, just tell me your name.
So hello everyone. I'm Simran Maratha from Central University of Rajasthan, working in the lab environmental. Microbiology and biotechnology. I'm basically working on microalga, not cancers or animal cells. So it was completely new for me, I would say, because I want to delve into the proteomics of algal cells, which is not so much new. But to learn the basics, I had to come here anyhow. And I would say the experience was ultimately good. The patience level of Graham sir was heads off, because it was not my cup of tea, I would say, machine learning and AI, but the way you handled all of us so patiently was again remarkable. Thank you so much, sir. And one thing I would like to mention is that in last few years, after I completed my masters, I haven't seen anyone as good in teaching as Arup sir. Like the way he engages us while teaching, he doesn't even need mic or anything else, but hats off to you, sir. The way you teach is completely remarkable, and I would love to learn something else from you also. Every time you will organize something, I would like to join.
Right, I hope I get giving funds.
One thing they both mentioned was, Graham sir said that biology is an orchestra, which I like the most, because we learned everything here: mathematics, computational science, biology, everything. And Arup sir said one more thing, that there is never too late to change. because he jumped from genetics to proteomics, so I might think of jumping to somewhere else also in future. I hope I won't need it, but maybe.
You should keep changing with the times.
Yeah.
Never be afraid to reinvent yourself. I was an ecologist once.
Okay.
So.
Thank you so much, sir.
Thank you, Simran.
Thank you.
So, hello everyone. My name is Jeevan and I am from Allahabad, currently working in pharmaceutical analysis department. And I was majorly here to, I'm already facing challenges in my research work. So the things that I've learned here, maybe I will go back and implement those. And I've learned like every bit where can I go wrong and what needs to be corrected. And I enjoyed all the lectures very thoroughly, and especially Arup sir's lectures were very, very engaging, and the information provided was it was very much I have read everything from papers and framed my project, but that whatever I've read, I came to know in the form of lectures over here, and that was very much informative for me. So it was a very nice experience. And I would also like to join more this kind of programs if it is happening in future. Basically, which includes proteomics, because it is a very new field and emerging field. People are trying to work in India. So I'd love to join more sessions. So thank you so much.
Thank you.
For having us.
Thank you, Jinal. And yes, you can. You can look at the PSI website. They keep on, advertising lectures. You can also look on the HUPO website. There are multiple webinars, and also multiple companies like Thermo Fisher, Brooker. They on their own websites they have tutorials. Like there is a very nice website, I think Orbit Trap, something like, I'm forgetting the exact domain name. But if you Google Orbit Trap website, they will take you to a website where they have lots of videos and animations. So time was one of the things that we could not manage well, because we had to tell you so many things in so little time. So you can go back home, look at those YouTube videos where you will understand how a hybrid mass spec works, how a tribrid system works. There are good animations. And now that you have the basics right, you would follow the animations. That's the more important part.
Yes.
So,
and again, look at the videos for Weka, for Orange. There's a lot of material there. There's a lot of depth there. I would also say there's an APT meeting, which I think has a workshop in initially as an IITB, then we'll move to BHU.
yeah, so there are two areas. I think in APT they are having 2 workshops, and if you want to get into very detailed workshops, you can go there, APT 2025 in IIT Bombay, and then even I'm part of this BHU workshop, but that will be more on the basics. So BHU workshops are more if you will learn more about the basic part of proteomics. But since I think you have already done this GIAN course, you might attend. I'm not asking you not to attend, but maybe you can also find, because APT has a much more, there are I think 4 or 7 parallel workshops that will be going. And if you want to do maybe cell surface proteomics, they have a separate workshop there, and people from really big institutes like Harvard and UCSF, people who are standardizing their own techniques, single cell proteomics. We did not discuss a lot of stuff here because, again, the idea was to bring you up to speed to where we are. And now I think once you have the fundamentals right, you can learn all by yourself. There is lots of, Coursera is a very good, Coursera, you can audit a number of courses on Coursera.
Yeah.
You won't get a certificate, but I think more than certificates, what you really need are skills.
Yeah.
Learning this.
Skills will beat a certificate any given day.
Yep, definitely.
Okay.
Good afternoon, everyone.
Sorry.
Good afternoon, everyone. My name is Garima Gupta. I am pursuing my master's from University of Allahabad. And first of all, I would like to thank Professor Graham sir and Dr. Arup sir for conducting such an interactive and informative workshop. Am I audible?
Yeah, you're audible.
May I just say like that only, sir?
Yeah, you can. Yeah, fine. You can use this mic. This is less sensitive.
Hello.
Yeah, you're audible. Yeah, you're audible on the mic.
I'm audible on the mic.
Yeah,
Am I audible on the mic?
you're audible everywhere.
So, I can just mention one unique thing I learned here, because everything I learned here was unique and new for me. And the one specific thing was that orange app that I learned here. It was new for me. I thought it might be very easy to use. I just went and I downloaded it, and I failed at putting that input CSV file. And then I came back and I was like, sir, what's wrong with it? And sir guided me that yes, you can do it like this. So it was very nice, and that Shambhu sir thought that metabo analyst, that was also new for me. And I personally feel that it was a very nice workshop, because I found myself interacting with so many people, so many good people. Initially I did not had any idea of what PhD is like, but now I know. I got a glimpse of it, that what we exactly do in that. So that was a very good experience.
So I hope you are pursuing a PhD.
Yes, sir.
You mean it in the positive way.
Yes.
So they didn't.
And personally, since this workshop has inspired me to attend many more workshops and gain knowledge about more research and PhD. It's really fascinating and interesting to do.
Thank you, Garima.
Thank you.
Hello everyone, myself Nikum. I just completed my PhD from the University of Allahabad. I worked on the integrative omics, and earlier I was planning to work on the ML. But I went through lots of tutorial and article, but nothing worked for me. But Sir guided me, and all the presentation that you showed, so that enlightened me, and I already started work on this. So thank you, sir, and it might be helpful for me, and I contacted you in the future.
Yeah, if you have any questions, please feel free. And I'm always developing new techniques.
Yes.
So if you speak to me in 6 months' time, I may have a new idea to try out as well.
Okay. So good afternoon, everyone. I am Fiza Fatma, and currently I am pursuing my PhD from IIT Allahabad, and I am working in the IRCD lab right now, which is Indo-Russian Center for Biotechnology. So there my research interest is antimicrobial resistance, and I was very new to this proteomics thing, but from here I am taking a new idea to initiate it in a project. So thank you so for such an engaging and interactive discussions.
Thank you.
Thank you.
Hello everyone. Myself Vidushi, and as Mr. Daman said, I am also working in IRCD lab under the Dr. Sindhu Samanta. I'm already working in bioinformatics fields, but for now, proteomics is new for me. I'm already working in bioinformatics field, but in the evolutionary field. But this proteomics thing is new for me, so I gain a lot of knowledge in the field of proteomics. So thank you, Yaman sir, and thank you, Karan sir. Like in this age, they have done so many activities. It was good for me. Thank you so much, sir.
Thank you.
Hello everyone. I'm Shivani Chandra. I'm from University of Allahabad. I'm in MSc second year. I was new to the subject, but the information provided by both the sirs was very informative and has improved me to explore more about the subject. And I'm also thankful to them for taking our doubts and questions and giving us quite satisfactory solutions. Also special thanks to Arup sir for providing us yummy food. Thank you.
Yes. नहीं अंदर बैठा उनको शेयर दे दो.
My name is Neha.
हाँ लगाओ पीछे से.
I'm pursuing MSc Zoology from University
लगाओ
of
लगाओ.
I would thank both Ram sir and Anup sir for this workshop. Even though this topic was new for me, but yesterday I was able to follow the tools that was teached by Ram sir. And also I would also like to thank Shambhu sir because he was very patient while I was asking him doubts about meta-analysis. Thank you.
Hello everyone, my name is Pratibha Sarosh and I am a student of MSc second year from here. I would like to first thank Arup sir and Ram sir for this interactive sessions. Sir,
हाँ लगा दो तीन
this
लेकिन
proteomics
नेम
and
प्लेट नहीं है ना
AI
तीनों का. दो ही चेयर लगाओ.
is very new thing for me and I would like to thank you. And one concept of proteomics and AI that I am interested in is digital twin. In my future, I would like to
चार बजे आ जाएंगे.
studies in digital twin and I would like to test the toxicity levels in comparing the a normal body which corresponds to the toxicity testing. Thank you, sir.
That's a really good point because nobody's really done that yet.
I'm highly interested in that.
So I would just request you, I mean, as much as it is really flattering to hear good things about us, but we are running out of time. So the worst thing is Professor Rizvi arriving and we are still continuing the discussion. We have 20 minutes to wrap, around 20 people to go through. So be brief, maybe you can talk about one point, and I think both me and Professor Graham, by this time we are convinced that we are super awesome.
It's like head was growing like this.
So the way things are going on, maybe we will explode with happiness, which we do not want to.
It's very messy.
So maybe you can just stick to one topic that you really loved once.
Sir, I would like to add something that challenging things.
I will take this. To overcome the fear of failure, keep failing.
Yep.
Keep failing so much until it stops hurting you.
Yeah. Yep.
But even when I started off as a PI, my first research project got rejected for funding. I was really upset for a week. Just before GIAN started, one of my research projects did not get funding and I didn't even care.
Yep.
I was like, okay,
Whatever.
so keep failing. Keep trying, keep doing new things,
Yeah.
keep failing, and one day we'll succeed. And all of us will have almost a 50% share of failure, in fact higher than that.
Yep.
And we succeed in only about 10 to 15% of the stuff we do. And a smart man will only show his success to the world. But a wise man will tell that he has failed more than he has succeeded.
It's what you learn from your failures that is more valuable. What you learn from your failures is more valuable than what you learn from your successes.
So,
Thank you, sir.
thank you. Ramjim, you have 30 seconds.
Hello everyone, my name is Ramjan Srivastava and I'm also doing my second year. I'm actually second year. So I think everyone here is new to the field of proteomics and AI and I'm really willing to get into this more deeper.
Hello, my name is I'm from Orissa. Currently doing IMSc in second year. I would like to, this content was very clear. I like to thank Ram sir and Arup sir. And I like to handle this type of workshops.
Thank you.
My name is Sumudupal. I am currently pursuing my Masters in Microbiology from Ramakrishna Mission, Vivekananda Centenary College, Kolkata, West Bengal. In masters, this proteomics is totally new topic for me, but the way Ram sir and Arup sir teaches us, it likely I. Thank you.
I think what we asked was what you learned. We know we teach so well. Maybe one thing that you learned, something maybe you can learn.
We need to know what you've learned, because then we know we've actually taught it.
So maybe you can tell that I did not know that in mass spec you need to digest the proteins with trypsin, or we did not know that how the bioinformatics part of detection on the mass spec, how sequencing works on a mass spec, what is LCM SMS, we did not know. Something that you knew, right? We did not know what is how generative AI works, how explainable AI works. So some one topic, you have been taught so many lectures, so the rest part is fine. We know we teach really well. Thank you for being the patient student.
Good afternoon everyone, I'm Anshul Dubey from University of Allahabad and pursuing my PhD from Biochemistry department only. So my work is also on the breast cancer, but honestly I have no idea about the proteomics and even much less about the machine learning thing. I was very interested in the proteomics part, but about the AI part I was not sure. But from Professor Graham's lecture, especially he has shown most of the case studies of breast cancer. So I was very interested in knowing that we can collect the proteomic and transcriptomic data from the group taught from the PAGC website, and we can run those things. And the most important thing I found out that we can actually make a very good hypothesis based on those things. Like we still would need the validation to continue those things, but the markers that we would choose would be much more, because what I feel I've been doing was pretty much like this, even in my PhD, was this discovery thing. I induced the cancer, then I tested for my phytochemicals, but the preciseness was not there. So further going, especially in my PDF project, I would like to implement both of those things, and I feel very confident that these things are not new random terms for me. So I would be very happy and I would certainly continue and implement these things in my project. So thank you so much, sir. Both of you. Thank you.
Thank you.
Hello everyone, myself Priyanka Tiwari. I am also pursuing PhD from Biochemistry department here only, and I have done my PhD work on mice, and we see the corticosteroids induced toxicity in rat, and its and phytochemical based implication, how these side effects can be protected by the
हाँ सर हेलो आपके पास सर फाइनल जो लेक्चर है स्टूडेंट पार्टिसिपेट इंटरेक्शन
gone through the metabologist and orange light formula and also the sample how
बजे आप बोले थे तो 4-4,000 पे कहाँ जाइए ठीक है सर हम कॉल कर लेंगे ठीक आप नीचे आएंगे क्या अच्छा सर ठीक है सर मैं आपको बुला दूँगा जी सर कॉल
So I have some basic understanding of machine learning and proteomics here. Thank you very much, sir.
Thank you.
Professor Graham for providing this workshop.
Okay, great, thank you. There's somebody else in there.
Hello everyone, my name is Sandevi. I am currently pursuing my PhD in botany department. My background is physiology. This works are very new for me, proteomics and AI. I don't know about ABCD of AI. Here I am learn so many things about AI and tools which can use efficiently and enhance my work. Thank you so much.
Thank you.
Hello everyone, I am Kishore. I am doing my PhD from Allahabad Academy of Higher Education. First of all, thank you everyone for connecting me to it. And the most fascinating thing about this GIAN program was the various case studies which were listed, which helped me know how proteomics and AI
was being used for various problem solving aspects.
I've set a time also.
Hello everyone, my name is Gopal Dutta. Currently I am pursuing Masters in Microbiology from Ramakrishna Mission, Vivekananda Secondary College, Kolkata. I learned a lot of new things about proteomics, like Max Command, various tools and techniques. Thank you, Officer and Ramsar, both you are organizing such kind of things.
Thank you. Excellent, Gopal. You are
I'm from Amravati University.
right.
So, I'm from Amravati University, so that course definitely teaches us AI tools, how to use that, and by doing more practice, and by application of AI tools and machine learning methods, the question is arise that I will definitely thank you, sir, and thank you for having me here. Thank you, sir.
Thank you. Thank you.
Good afternoon, everyone. Hello, this is Yashmi. I'm from Eros Medical College, Lucknow. I've come from Lucknow all the way here. As I have been enrolled recently in my PhD in plant proteomics research. I've known quite much about proteomics, but there is a lot to know more, which I came to know here only. And interactive sections was quite interesting, and all our instructors were also so supportive, and they taught us in a new way that got to know about very new things. And it was a very nice experience. I've learned a lot, and would like to know more about it in future sessions. And thank you to all my instructors for helping me and guiding me.
Thank you. Sorry, but we have to keep up the time. Yeah, 30 seconds.
My name is Indrakshi Datta, and currently I am pursuing my master's from Baba Saheb University in Lucknow. The whole was new to me, and I think it might help in future to apply somewhere. Thank you.
Thank you. Excellent introduction.
Hi everyone, I am Neeraj, and I have recently completed my postdoc from Penn State, and I am currently I have recently completed my postdoc from Penn State, and my area of research is AI-driven drug discovery. So I have already developed few algorithms, and currently I want to you can say enhance my research domain or research area to proteomics, where we can fetch proteomics knowledge along with the structural bioinformatics, so we can have new tools and some better therapy and therapeutic details. So that is what thank you.
Thank you.
You guys are wonderful. Thank you. Thank you.
Yeah, that part is clear. That part is fine.
Good evening everyone, I am Siddharth Sharma from JCRC University, Jaipur. In this it was my first experience. I am pursuing PhD Honours Biotechnology from JCRC University. So it was my first experience of conference. So here I am glad to learn about the proteome and the softwares which are working about proteomics and
नहीं वैलिडेटरी के बाद जाएंगे.
Thank you.
Yeah, you're audible.
This is Udit Narayan Mohanlal from BJP Autonomous College, Bhubaneswar. Now I am doing my bachelor degree. I am in second year bioinformatics. So I don't know what to say, how to say, but I get to know more about like biological tools. I knew only NCBA, drug bank, but I get to know more. Orange, metabolism, symptom. Thank you.
Thank you. Thank you.
Hello everyone, myself Priyagraj
informative and very funny because of I am just passed the PG from bioinformatics. So in the future, it's a very knowledgeable for me. Thank you.
Excellent.
Hello everyone, myself Sneha from Orissa. I am going to my UG second year in the Department of Bioinformatics, and GIAN provided me a new software to learn about proteomics and its applications. It was a very knowledgeable.
Hello everyone, this is Ranika Sani from Center of Environmental Study, University of Allahabad. I am currently pursuing PhD and talking about PhD is basically based on absorption and degradation technique, which is completely different from proteomics and AI. So the way what taught us was really amazing, and the patience you have and solving you have was really amazing, and I learned many new things from this.
Thank you. There's some people who just come in, I think. So.
My name is Islam Muller. I'm from Jaipur, GIAN University. तो सर मैं आपको थैंक होना चाहता हूँ ग्राम सर को, आपको और ऐसा बहुत अच्छा आदमी आपका समय आप मुझे कॉन्टेक्ट बनाया, मेरे से बात की, तो मैं सर को थैंक यू होना चाहता हूँ। और मेरा फर्स्ट एक्सपीरियंस है, इसलिए मैं तो अभी तो BSc सेकेंड ईयर में ही हूँ, मैंने नहीं देखा था कि इतना कुछ भी होता है, प्रोटियोमिक्स में कुछ इतनी बड़ी चीज़ है। थैंक यू सर।
Thank you. I mean, what he told was he loved the course, and this was his first course like this, and he is in his BSc UG second year, and he did not know such things exist. I think our biggest hurdle is not knowing things that already exist, and that's a lot of discovery phase that you should do yourselves, and discover more what you do not know, and then perhaps you would find your calling, right? I discovered proteomics when I went to a conference in France, and I saw a 2D gel with lots of colors. So I did not know what it is, so I only picked up Daij, because that was mentioned on the poster. It was in the year 2014. I came back to India and I started looking up what is Daij, and that's how things begin. I mean, you travel somewhere, maybe in this old 5 days you would perhaps pick up Silac, or maybe some machine learning algorithm, or maybe you will pick up Weka, and who knows, 10 years down the line you will be, I mean, it's almost 10 years. 2014 I learned about Daij. In 2024, and after a decade, I'm organizing and teaching in a workshop on proteomics. So you never know where life takes you, so it's a good thing that you keep exploring other topics as well.
It's the lady in the pink top.
Okay, please.
I'm working on plant file isolation and identification characterization of Latin from predictive plants. Second was very informative on.
Thank you.
I think
that's a good thing.
The last minute volunteers. आप लोग कुछ शेयर करना चाहते हैं? Anyone? Anurag, come. Aajo. Yes.
So there should be some tools like cancers for homocities.
Aajo.
Yes, yeah.
Aajo.
For LP and phyto, I would say phytologists.
Yes.
There's nothing such data
Bolo.
Start working because biology includes everything.
If there's, and that's the thing, if something is not there, you are the first to do it. If you're the first to do it, that has impact. Yes. So making data for an area, analysing that data, and you're the first to do it, that has impact. Yep. So never be afraid to be the first to do something.
Because we are failures and assess that software failure.
Yeah, I mean, I certainly talk at lectures, and I'm not afraid of what people think when I say things, but I think everybody has a fear of failure or fear of things going wrong at some point.
I think research is 99% failure and that 1% success that we publish.
Yes, yeah. Or some people say 99% boredom and 1% inspiration. So that's the other bit. But yeah, I'm happy to reach out to things.
Currently, I was studying neuroscience, and I was just figuring out that what should be my topic of dissertation or what field I should pursue for research.
Yes.
And I figured out that cerebellum is the least studied organ.
Yes,
of the
yeah.
brain. So I thought we can do cerebellum proteomics. I can apply proteomics there.
Well, have a look at brain prot.
Brain prot.
Brain prot. So one of my former PhD students, who I co-supervised at ITB, has produced this brain prot, which is the brain proteome map. So that's a good starting point for you. Reach out to him. He's at Harvard now as well. Yeah, he's a good guy.
So we have friction from the volunteer team. Maybe they have done more.
Administrative part. And management, how to manage.
How to manage
Excuse me.
the friction period for all of us.
Good evening, teachers, and good evening to everybody present here. First, I would like to thank all of you who come over here because me being a junior, I am from MSc third semester. So I met a lot of new people
Look at him on
and we saw how my seniors are working in this field, how much so are dedicated and how much they are worried about their results not coming. So this was very highly motivating. And thanks to our teachers, Professor Graham, thank you so much. You took time. You came from so long for us. You cleared all of our doubts. And Dr. Arup, I would like, Professor Dr. Arup, I would like to thank you, thank you for everything. And also, I would like to say that we had only heard about ChatGPT in terms of AI. We did not know anything. We did not know how big this world is. We saw how we can collaborate our data with AI, how much AI can help us. What are the limitations of AI as well. We got to know this, and we are really thankful that you came here and you took time for us.
Well, also thank you very much for all your support, for helping me deal with exploding microphones, for helping me deal with exploding toilets, and many other issues. But thank you for sorting that out and helping.
It was our honor to have you over here, and sir, thank you so much for organizing this for us.
Thank you, Shamsuddin. So, who's that?
My junior is supposed to come, but like he got infected with dengue, so he couldn't come.
So anyway, you can tell him that somebody is using his time.
Today only I left it there at night at that venue.
Okay, so Shamsuddin is from Nagpur, right?
Yeah.
Okay, so I think we can settle down. We will now start our valedictory. Professor Cyrus D, who is the Dean Research and Development, will be joining us. And volunteers would like again ask for your assistance in setting up the stage. I think we did not have your calls.
You didn't. Nobody gets away with it.
No, sir, it's not you.
No, just want to look at the
I'm a third year research scholar in Department Center of Biotechnology here at Allahabad University, and I've already discussed with you a lot. So, like my work is in RNA therapeutics using nanoparticle mediated delivery. So, like this program has given me new insights and new perspectives how I can apply it in my study to modify like my area of research. So it will be very much applicable. So for any cross-reference and any discussion, I'll come back to you.
Please do. Please do. Thank you.
For allowing me to speak.
Thank you. Thank you. It's a dangerous weapon, this is, in the hands of an inexperienced.
Yeah, so, volunteers, we have 5 minutes to prepare the stage and we'll set up a call. seriously. Yeah, webcasting thing, you can continue with casting.
Different seat you can
So also, we cannot accommodate everyone's view on the stage. So one of you can volunteer and let me know who is volunteering so that I can call him or her out. So have you decided amongst yourselves like who is volunteering? Preferably somebody who is not from Allahabad. Because else it will look that I have influenced them at the gunpoint. It was really very favorable to new things. Anyone? Anyone wants to speak maybe 2 to 3 minutes? Simran Maratha wants to speak? So either we can go by consent or we go by random numbers. My students, if they are present here, so they might know. I usually generate random numbers in Google and call the roll numbers out when I am in the mood to take a test. I think I have done with these once or twice. So whichever way you want. But I think someone can use, you all spoke really well. So,
Yes,
and
yeah.
it's not something that, it's not an operation that all the top bureaucracy will come.
Yes, it's not.
Professor Rizvi is very friendly. He's also a good supporter of science, so not worry.
It's not being broadcast on Sky News or anything like that. Just gonna pop through.